So you found the skeleton. How would you tell people that you personally? How would you decide?
Well, interesting question. I don't know. I'd have to do some research on that.
Hello, everybody. Good afternoon for almost all of you, I think. Maybe good morning for a few of you. Dapper Dinosaur here. I am this time joined by Dr. Zach B. Hancock, who is an evolutionary biologist specializing in population genetics, molecular evolution, and phylogenetics. He is a postdoctoral researcher at the University of Michigan, PhD in ecology and evolution from Texas A&M. So, Zach, which is what I'm going to call you because it's what you put on screen.
Yep.
How are you today?
I'm very good, thanks. I'm excited to be here with somebody that just reached 10K subscribers. Congratulations.
Thank you very much. Yes, my plan is to have a celebration stream about a week from today.
Awesome.
By the way, we have a one quick super chat for €2 from Pelagic Tunicate Truth Official, which is one of the greatest names. So just a reminder, the Pelagic Tunicates love Dapper, which, I mean, they might, but the feeling is not mutual. I don't know if you know, but this, at least on this side of the camera, this is an anti-Pelagic Tunicates. We're much bigger fans of the sessile Tunicates.
Oh, okay.
Yeah, we like those guys, because those are good, hardworking, God-fearing, patriotic Tunicates. And the pelagic ones are heathen, godless, unpatriotic layabout. Well, not layabouts because they swim about. Swimabouts. There you go.
So do you have strong opinions about the parasites of pelagic tunicates? There's a amphipod that parasitizes pelagic tunicates.
I am not at all surprised to hear that there's an amphipod that parasitizes pelagic tunicates. Is there anything in the ocean that doesn't have an amphipod parasite?
That's a good question. Amphipod or isopod? Isopods parasitize probably even more things.
Well, which brings something up. So your field of research is heavily crustacean focused, if I'm not wrong, right? You just recently got back from collecting crustaceans in the field for your research.
I did, yeah. I study beach-dwelling amphipod crustaceans. I have taken calling them beach moles because every study system should have a common name, and hosteriid amphipod makes people's eyes glaze over. So I've taken to calling them beach moles, and they live on sandy beaches. If you like going out to sandy beaches on the open coast, you can dig them up. If you've got like a kitchen strainer, you can just sift the sand and you can collect them yourself. They're all over the beach, really cool little critters.
Awesome. We got one more super chat for $4.99 from Vandalia1998, who says, "Got vertigo, so laying down before my stream with Bede. But here's some therapy money if you have to deal with another young Earth creationist." Thank you very much, Bede. I hope you feel better. All right, well, so let's talk about the reason that we're here. Because, so, backing up a little bit, I had done a series of videos in which I critiqued John Sanford, not primarily about his genetic entropy idea, although it did come up. Although you might not know that if you, say, hadn't watched the series and just got angry and tone policed me instead. Might not realize what was actually in the series if you did that. No particular reason that I might be bringing that up. But anyway, in response to this, one Sal Cordova, who is, as far as I'm concerned, currently still a friend of the channel, who's been on this channel more than once. I've been on his channel more than once. Decided to do a response, curiously, without having watched them. Now, the first response was just, I've described it charitably as hagiographic. I've also used more colorful words when I'm in private. A particular word derived from a Latin verb starting with F has been thrown around in there. But anyway, that was basically just how much of a big brain John Sanford is. And quite frankly, I'm happy to say that John Sanford is a gigantic galaxy brain, because the smarter John Sanford is, the harder it is to excuse his woeful inadequacies in his theoretical work as just him being bad at his job. And the easier it is for me to say, yeah, I don't see how this could be anything but dishonesty. Now, there was a third response, which was just, DNA folding exists, therefore evolution wrong, which doesn't follow, and I couldn't get anything other than, it breaks the laws of physics without any description of which laws of physics were broken and in which way they were broken. So I'm opting to ignore that until an argument is presented. However, in the middle, between these two, was a rather It was a somewhat unhinged rant about genetic entropy and why it's totally real. Trust us, you guys. And so we're going to talk about genetic entropy. We're going to look at that video. We're going to also look at a paper that was used by Sal in a... I'm not sure how to describe it. It was in a manner that I would say would not be acceptable in most academic circles if you used a paper this way. Namely by cherry-picking not even just the abstract, but part of the title, as if that phrase in and of itself stood alone and could therefore be used as some kind of argument about evolution, which I think is a rather shady. It's a shady way to use that paper.
Yeah, it's an interpretation that the authors obviously would absolutely have disagreed with.
Oh, yeah. So we had a couple of things here. So Hipplepunk says, hooray for 10K. Here's to more milestones soon. Thank you very much. That was for $2 US. Now, Alia Jane, who is a member and has been at the Sauropsid level for, I believe, 6 months, says, Eoraptor is my favorite dinosaur. And then also sends a Godzilla and a Pelagic Tunicate emoji and a trans rice. Yes, trans rice. Also, Alia Jane, every time I put you in my credits, I have to guess how you actually spell your name now, because now you have your name put in here in the International Phonetic Alphabet, which most of my graphic softwares will not interpret correctly. So it's always like, it's because I tell my audience, like, hey, if I ever get your name wrong, if you know the International Phonetic Alphabet, you can put it in that, and then I will almost certainly get it at least very close to right, because I'm pretty familiar with the International Phonetic Alphabet. But then, then she just changed her YouTube name to that. So, I love it. So let's go ahead, and I want to get some groundwork laid. Now, you being an evolutionary biologist, I think you're pretty much the ideal person for me to ask these questions to. Let's start with some basics. What is fitness in this context of evolutionary biology?
What is fitness? So fitness, there's several different ways to think about fitness. There's a short-term and long-term fitness, but I think we can kind of simply think about it as the contribution of an individual to the next generation. Mm-hmm. So relative to other individuals' contribution to the next generation. I like to think about the population as like a giant pedigree, right? That everybody is connected in this pedigree in a sort of a tree-like fashion. And every single generation, individuals leave offspring into the next generation. And the number of offspring every single individual has increases the genetic contribution to this whole tree, right? Mm-hmm. Across successive generations. So the more that you leave, both in the short term, so one generation at a time, influences your short-term evolutionary fitness, and then your contribution across many generations is influencing your long-term evolutionary fitness. And so these are sort of the concepts, just simply and broadly speaking, that encompass fitness. Now, there's complications in fitness in that, and maybe I can wait until Sal gets into it, but that's sort of the general. I'll leave it there and say that's sort of the general way of thinking about fitness.
Okay. Now, I think we both know that the people who are proposing genetic entropy as some kind of gotcha against evolution, some kind of problem for the science of evolution, they often will refer to fitness in a different way. I'm going to talk about my understanding of how they do that, and then we can talk about perhaps why that is not the best way to think of it. After I read a quick super chat, this is for $5 from Too Young to Feel This Old, who says, always glad to see a new face and be introduced to a new channel through Dapper. I will be subscribing shortly. Hashtag during the Pelagic Tunicate Revolution. That's a very long hashtag. Yes. Zach's YouTube and his Twitter handle are both listed in the description, as well as Sal's YouTube channel. So if you want to go watch his responses, generously so-called, feel free. I watch them. I do extend that courtesy to people who I intend to respond to watch the thing I intend to respond to. I know it's a wild world we live in, where people might respond to things after having watched them. Okay, so the advocates for genetic entropy, from my understanding, tend to define fitness less as to do with things like reproduction and the spreading of one's genetic, say, distinctiveness into subsequent populations. That sounds too much like the Borg. Add your biological distinctiveness to the population. I guess it's accurate, right? Like, it's not wrong. Anyway, and instead they like to define fitness the way that, like, say, your personal trainer might. Like, how much you can lift, how fast you are, how good your eyesight is, I don't know, how good of a beard you can grow. Any of these things might be called fitness in this sort of broad sense. Now, why is it that that is not what we normally talk about in the field of evolutionary biology? Whereas, I mean, we might talk about it in other fields, right? Like if you were trying to assess, like, hey, what animal can jump the highest? By the way, it's the pronghorn. You would talk about that, right?
Yeah.
Like the muscle strength and all this cool stuff. So why doesn't that work so well in evolutionary biology specifically?
Yeah. So I mean, just to like think about like what evolutionary biology is interested in, we're fundamentally interested in the change in the heritable characteristics of populations across successive generations, right? And so the fitness that we're typically interested in is the fitness that is contributing to that change, right? Now, individual traits may have, may be contributing positively, negatively, or not at all to that specific change. We're specifically just interested in the heritable variation because that's how we trace evolution, right? Evolution is fundamentally linked to heritable variation. Now, your personal trainer there may be some component of heritable variation that your personal trainer is concerned with, but you don't pass on your individual muscle mass to your offspring, right? Like that's not an evolutionary measure because it's not heritable. It doesn't continue to get passed on. And so it's not a measure of fitness that is very useful because it disappears every single generation. Since evolution is concerned with the changing frequency, the changes in heritable characteristics, we need to focus on a definition of fitness that can actually track those heritable changes.
Now, let me do a little bit of devil's advocate and say, okay, but what about, say, something that is strongly heritable? So, for instance, let's say that there's a, maybe it's a metabolic pathway, right? So I've got this metabolic pathway, and my alleles for the genes involved in this metabolic pathway result in a very high efficiency metabolic pathway. Someone else's result in a much lower efficiency metabolic pathway. But we'll say that in this context of my population, this metabolic pathway is actually not under any particular selective pressure. Maybe this particular thing that I'm breaking down just isn't prevalent in the environment, so I don't get a lot of it. So there's very little advantage or disadvantage. Now, the proponents of genetic entropy would, and I don't think I'm strawmanning here, I think I am accurately representing them, would say that my version of the higher efficiency is fitter, and the lower efficiency is less fit. But in evolutionary biology, if we say, well, there's no effect on reproductive success, whether in the short or the long term, then we can actually say that those two different genotypes have the same fitness. So why is it that what I'm describing, where you just have different efficiencies of, say, some metabolic pathway that isn't under selection, why doesn't that count as fitness for this discussion?
Yeah, because the allele trajectory of each one, so assuming it's like a simple biallelic trait, the allelic trajectory of those variants is determined by random genetic drift. Okay. Right. So if they're not actually contributing to differential reproductive success, such that selection can differentiate between those variants, then they're predicted by just stochastic drift. And so in that case, there's no to be more fit doesn't actually mean anything. It doesn't change whether it's going to be more present or less present in the next generation. And so they're going to have to come up with like a meaningful definition of fitness in this case that we can actually like make a prediction, right? So with evolution, if these two things are selectively equivalent, then we can make a prediction about how they should change in the frequency across generations. But if we go with their definition of fitness, we still have no idea how they should change, right? It's very unclear about what these two things should do.
Yeah, that's one thing I really want to highlight, is that one of the most important ways that you can tell if your idea is something that science should take seriously is if it, one, makes predictions, and two, those predictions are actually confirmed. And I think one of the things we might get into later is whether or not the predictions that you could make from genetic entropy are confirmed outside of computer programs created by the people who like genetic entropy to result in genetic entropy happening in the simulated populations. Yeah. Because I'm going to throw that out there. I don't think that's a very fair test. That's just you creating a program to get the results that you want and then being shocked and amazed that, oh my goodness, the computer program that I wrote to demonstrate genetic entropy just demonstrated genetic entropy. Like, well, yeah, that makes sense. I would expect it to. All right, so I think at this point we can jump into the show that Sal put up, the response, if you will. And what we can do is, whenever you or I have something in particular to say, if I have something, I'll just pause it. But you can always tell me to pause it at any point. We'll go over whatever he's got for us. So I think we've... Oh, there's one more thing I want to go over, which is deleterious or beneficial versus... beneficial or deleterious versus and harmful. Like those two things. When something is deleterious, what do we mean?
Yeah, so there is two different ways to think about this. And it actually kind of gets into the heart of the history of population genetics. And I think it's an interesting discussion. So I'm going to take just a little bit of a detour and try to explain it. So you can model the change in allele frequencies either deterministically, or you can model them taking into account stochastic effects. Deterministic models tend to be like kinematic expressions, so they're analytical. They have variables, you plug in numbers, and poof, it gives you an answer, right?
Yeah, it'll always be the same answer every time with the same variable.
Yeah, exactly. It will always give you the same answer, and it makes a very predictable curve. It gives you very clear answers. In those cases, if that's the way you model your population, when we're thinking about what's beneficial, deleterious, or neutral, what we mean is just the sign of selection. So is selection positive, negative, or does it equal 0, right? And in that case, the allele trajectory is very deterministic. Now, it's a little complicated in diploids because then it matters what's the coefficient of dominance, right? So if it's dominant, is it additive or is it recessive? If it's recessive, for example, then alleles, even if they're positively selected, don't ever go to fixation in deterministic cases. Because the alternative allele, the less fit variant, gets hidden in heterozygotes. Right. So as it gets really, really, really high, it'll get high really fast, and then it'll just like sit at near fixation and never quite get there. But again, that's in a deterministic model. So we have a very clear definition of what's deleterious and beneficial in that case.
Now, beneficial is for a positive sign.
A positive sign versus a negative sign for deleterious, exactly. And so then actually like for selective neutrality for 0, then the alleles don't change. That's effectively Hardy-Weinberg, right? For a deterministic model. Now for stochastic models, we're taking into account finite sampling effects. So we're actually going to consider what we call genetic drift. And genetic drift is just the stochastic sampling of gametes every single generation. Because you can't sample a population infinitely. So in the deterministic model, you're assuming basically an infinite population. You're not having to worry about sampling effects. Whereas in a stochastic model, you are. So now in these models, you very rarely get analytical, it's not analytical, so you don't get an actual value. You get a probability distribution. Right. Because there's some chance involved. Now in these cases, the distribution of fitness effects, if you can think about fitness as sort of a distribution with some mean that represents like this is selective neutrality, above that value is positive, below that value is deleterious, is negative. There is some population size that will then dictate how deleterious is. So for example, what is deleterious in a population of size 10 billion is very different than what is deleterious in a population size of 100. And what we mean by that is, does the frequency of that variant, is it dictated by selection? So is selection able to drive that frequency, or is it dictated by randomness? And for small populations with sufficiently small selection coefficients, they can't actually distinguish them, and so they drift to either loss or fixation. So in those cases, we call those effectively neutral.
Okay, and I think that actually ties into a question that Andrew Cumming had, which is it possible to have a slightly deleterious mutation that isn't selected against, or is it inherently a contradiction? And I think that ties into the population size, where you could say, well, if there were a population that were, say, 10 billion, then this particular allele would definitely be selected against. But if there were a population of 500, it has the same physiological effect on, the actual phenotype of the organism, but it might not be selected against simply because there's too much drift happening to actually have any noticeable selection against that particular.
Yeah, and there's like a simple rule to this. It's an inequality of 4 times the effective population size times the selection coefficient, and that value is either greater than or less than 1. And so if it's less than 1, then drift dominates. If it's greater than 1, selection dominates. And that's just like a simple inequality to help you know which one of those dynamics are going to dictate it. So you can see there's a clear relationship between population size and then, like what's the strength of selection.
Right. Okay, well, I think that actually does cover for real everything I wanted to cover before we dove into this. So now that we have that background, let's take a look at what Sal has to say. Now, I do want to say, this is something that Sal recorded a while back, I believe years ago, and it is not terribly representative of the work that he is currently producing. So it is a bit, I'd say it's a little sillier than what he typically does now, I think is a good way to put it. Frankly, it's a little snarky, but I don't really care. I can be snarky too. But it's a little silly. Like, there's a lot of, like, cartoonish graphics and things and, like, sound effects. And that's a little weird, but it's not representative of the current stuff he's producing. So be aware that this is, I think, probably 4 or 5 years ago version of Sal. So. All right, here we go.
Why have we not died 100 times over? Why have we not died 100 times over? This rhetorical question was the subtitle of a paper by geneticist Alexei Kondrashov in 1995. His 1995 paper highlighted a growing suspicion that the human race is decaying genetically and possibly headed toward extinction.
Is that actually what Kondrashov's paper was about?
That's not an accurate representation of what Kondrashov's paper was about. Kondrashov's paper was introducing the idea of drift load. Actually, I should be clear, it wasn't even introducing the idea of drift load. Drift load was a concept that had been around for a long time. And maybe I should define what drift load is. So for small populations, and we kind of touched on this a little bit before, actually. So for small populations, they can harbor more deleterious variants because selection is less efficient, right? And so what ends up happening is, by chance, in small populations, some of those deleterious variations can reach higher frequencies than what they would in very, very large populations. So we call this the drift load. And if you consider every single deleterious variant as additive, so like you can just add up the selection coefficients against every single one of them, and that summation represents the decrease in fitness of the population, right? That's what we refer to as the drift load, right? And what Kondrashov was showing is that there is a range of parameters in which organisms can accumulate very, very slightly deleterious mutations, assuming, again, the additivity of these deleterious mutations, and then making a few other series of assumptions specifically related to what's the relationship, for example, between the functional fraction of the genome, the selection coefficient against each one of those variants, and then the effective population size. So the actual go ahead.
Oh, real quick, I just want to make sure that we get functional fraction defined, because I think that's a new term. Which, now correct me if I'm wrong, but that is the fraction of the genome that is under selection and therefore can be reasonably said to be functional for that organism in the current environment. Yeah,
that's the fraction of the genome that if it mutates, if a mutation occurs in that fraction of the genome, it is more likely than not to harm the fitness of the organism. That's I think a key way to sort of think about it. And yeah, like selective constraint is another way to consider it. Selection is actively maintaining that region of the genome.
Okay, so one of the things I want to point out about that is that that definition of function is different than what you might think of it naively coming in, right? So you might think, like, I'll actually use an example from, like, human biology, right? So I think we all know that humans have a pseudogene that in other mammals makes vitamin C. It's the reason humans can get scurvy. But given that this thing managed to reach fixation, it's reasonable to think that in whatever organism this originally broke in, because originally, like, this mutation that broke it would have happened in an individual, right? Mm-hmm. That in that individual's population, that gene was not functional under the definition of functionality that has to do with things like selective constraint, because the very fact that it was of such little consequence that it actually reached fixation means that essentially a quote-unquote functioning vitamin C gene wasn't functional in the actual organism. And so you can have an organism with things that under circumstances might be an important gene that still has the same ability to function as it does in relatives. But because of that organism's environment, we can treat that section as non-functional for the purposes of modeling and for predicting whether or not it might break in the future or what might happen to mutations to it in the future population.
Yeah. I think a good way to think about it is it necessary? Right?
Yeah.
Like for vitamin C, it's it has a quote unquote biochemical function in that it does produce a product, but it's not necessary for the survival of the organism. And if it's not necessary, i.e., it's not being selected to be maintained, mutational processes will degrade them.
Right. Okay. I like that example because it's something that I think most people are aware of. Like, everyone knows, I think, that scurvy is a thing you don't want. It's a bad time.
Yeah.
I mean, unless you like having your gums bleed and then you die. I personally would opt out, but then again, being a dinosaur, I don't have to worry about these silly concerns.
That's true.
All right, here we go. Let's get a little bit more salad.
Then, in 2017, Kondrashov followed up his 1995 research paper with an entire book entitled The Crumbling Genome. Other scientists, to varying...
Now, I'm actually not familiar with this particular book. Have you read this book?
I have not read this book. I will say that Kondrashov is actually... his office is right there. Oh. He's right above me. I run into him on the elevator sometimes. So he is at the University of Michigan. And one of these days, I'm going to get brave enough to talk to him about some of these things. But he has this like old Russian demeanor to him that is like kind of intimidating.
Okay.
But one of these days, we're going to sit down. We're going to chat about this. But there's a few things of interest here. So Kondrashov is basically making the same argument that Muller made, and what a few other geneticists have made throughout history, that there is a mutational load in humans. And that's, from my understanding, again, I haven't actually read this book, but from my understanding, that's what this book is focused on. I do want to add that it is also Kondrashov who, just a few years after the publication of this book, showed a way of escaping the mutation low, what we call the mutation low paradox, which Sal will get to shortly, by what we call synergistic epistasis. And he was actually on the paper that found empirical evidence for synergistic epistasis, okay. Which I can go into, or we can get into a little bit.
We'll see if it comes up, but would it be fair to say that, to your knowledge, Dr. Kondrashov currently does not espouse the views put forth in this book because he has found a solution to the alleged problem?
Ooh, I don't know that. I don't know.
Okay,
that's a good question. I could ask him, and I will ask him, but I don't. Yeah, I don't know for sure.
Okay, hey, fair enough. That's much better than you speculating.
Yeah,
as if maybe, I don't know, you had seen a headline or a video title that you didn't like, and then just speculated as to what the video was about.
So I will say, like, a lot of this sort of thing, like The Crumbling Genome, there's another one that Michael Lynch put out called, it was like Human Exceptionalism in the Journal of Genetics. A lot of these sorts of things are focused on medical intervention in humans. So it's not even, so like what genetic entropy is focused on is not medical intervention. It's that naturally mutations will run us away, right? A lot of what these sorts of things are focused on is that medical intervention in modern humans has circumvented selection, right? So it's a different argument than what genetic entropy is arguing. Now, I don't think that that's true. I think that that's a misunderstanding of like the distribution of fitness effects. I think a better way to think about it is that humans have changed their environment. And so like the mutation effects are like, for example, I wear glasses. And so like in, 10,000 years ago, that might have cost me, fitness by wearing by having poor eyesight. But today I can wear glasses and I get by just fine. And so what we've just created an environment in which I'm not being hunted by big cats, right? And so the changing environment shifts the distribution of fitness effects. And I think that's a better way of thinking about how we get out of these problems than what people like Lynch and Kondrashov have suggested.
Okay. So there's a couple things. First, I want to read a super chat. This is from Vandalia 1998 for $4.99, who says, "You would think a non-functioning vitamin C gene wouldn't get passed along to descendants, but no one said natural selection was logical." Actually, it is kind of logical because the group of organisms in which this mutation occurred, which is common, it's very widespread in primates. I don't remember the exact distribution, but I believe it's at least all of the anthropoids. So that's like the New World monkeys, the Old World monkeys, the apes. I don't know if it's wider than that, but I know it's at least shared in that group. That's a group that gets a lot of vitamin C in their diet. So who cares if you're not making your own vitamin C, if you're constantly chowing down on leaves and fruit that are high in vitamin C? You don't need it.
Yeah, I think we should have brought that point out when we said whether or not it was necessary. Yeah, it's not necessary anymore because their diet shifted.
Right. Primates are primarily descended from insectivorous animals, which is basically true for any group of mammals you care to name, because primitively mammals seem to have been insectivores. But you need some extra vitamin C if all you're eating is like termites and ant larvae or something, right? Because they're not super high in that. But then you start eating fruit and leaves, and there's vitamin C all over the place.
Yeah.
So let's see. The other thing I want to point out, I really want to highlight that the argument here about humans allegedly removing themselves from natural selection as a result of things like medical technology or increased food production ability, right, is not the same as genetic entropy, because that is specifically something that is supposed to be inevitable in all populations, no matter what.
Right. Yeah.
So saying, hey, this one population has done a weird thing with their, with natural selection, even if it's true, does not help the argument of genetic entropy, because genetic entropy is supposed to be a universal law of genomes.
Yeah.
So even if Kondrashov stands by all of these conclusions, I don't see the point of putting this book in here, unless Sal failed to understand it after having read it, which I'm going to assume that, unlike my videos, that Sal has read this book and understands it, because to do otherwise would be inappropriately dishonest.
Yeah, I think that might be giving him a little more credit.
Well, I have faith that Sal would not cite a source that he has not read, just like I would have expected him to not respond to a video he had not watched. Now, my faith in that is starting to wobble a bit as of late, but as of now, I still have faith that Sal would have read this book. But we'll move on.
Degrees have raised similar deep suspicions that we are indeed de-evolving. That is to say, each generation has slightly more birth defects than the prior generation.
I want to say real quick, de-evolving sounds like going backwards, and that's not what anyone's talking about, and it's just a weird way to put it. That's all I wanted to add there.
The only debate is the severity of this decay, its causes, and possible cures, if any.
Okay, now, one of the things that I find interesting is that we don't have a citation of the rise in birth defects, nor do we have a good definition of what a birth defect is in terms of fitness. Because one of the things is, you could say, okay, Zach, you're wearing glasses, like you already pointed out. Is that a birth defect?
So, yeah, it seems to me like he's saying mutation equals birth defect. It seems like he's lumping in all mutations as birth defects. And given, like, if you think about the long-term effective size of humans as 10,000, right, which is about how much diversity we have in our genomes, is a population of size 10,000. And given that we have just exploded in population size, we should absolutely expect to see an increasing number of mutations, because you have more genomes to mutate, right? And this is standard basic pop gen theory. The bigger the population is, the more mutations that the population is going to harbor. So I would 100% expect to see that mutation should be increasing in the human population, just as, because it scales with population size, right? But in terms of their fitness effect, that's another component, right? It's very difficult to know their fitness effect in humans. We can do this very well in experimental systems, so like fruit flies, yeast, E. coli. We have good measures of their fitness effects. In humans, since we it's unethical to put humans into a lab, into a common environment, and like remove selection effectively is what you have to do. You have to inbreed and remove selection from these experimental populations to be able to see all their deleterious mutations, and what we call mutation accumulation experiments. But you can't do that in humans. And so it's difficult for us to actually know the number of deleterious mutations in humans. There's a few like little statistical and quantitative tricks that you can do to try to figure it out. But it ranges from like 1 to 10 deleterious mutations per generation. It's in that, generally in that range.
Okay. So a few things here. First of all, I want to point out that all these ethical rules were made up by the humans in the first place. So of course they have all these ethical rules. They didn't ask all the fruit flies or the rabbits about the ethical rules for them. Just putting that out there.
Yeah.
As a dinosaur, I don't have to worry so much about that.
Yeah.
Anyway, now you said that we do have good measures of these for a lot of laboratory model organisms.
Yeah.
And I want to once again hammer home that if this is, as Sanford is proposing, a universal rule that genomes all decay like this, it shouldn't matter if we're studying a model organism or humans. Their genome should be showing the same effects, and they're not.
Correct. Yeah. The distribution of fitness effects is different by organism. It's also a thing that can evolve, which is kind of interesting in and of itself. And it's related to environmental change. But it's also all of the genetic entropy assumptions is that the beneficial mutation rate is effectively so small that it can be ignored. And that's just preposterous. There's no empirically estimated distribution of fitness effects that doesn't include beneficial mutations. It doesn't have like a non-negligible number of beneficial mutations. So I think if you were to run a simulation, let's say, as Sanford and them do, and you only allow deleterious mutations with no compensatory mutations ever, then like obviously your population is going to collapse. You haven't actually simulated a real empirical distribution of fitness effects, right? But for all of these systems, or every single one of these model systems that we've been able to empirically estimate it in the lab, they include a sizable proportion of beneficial mutations.
Okay. Now, Titulak Rosi for 50,000 Vietnamese dong, I believe, is the currency. There's a lot of currencies out there. Did you know about this?
Yeah. There's quite a bit.
So it says, Salas Soblivy said he doesn't get it when people call him a hack fraud straight to his face. Also, I asked him, which kind is Titulak in? Nice. Look, I'm not calling Sal a hack fraud. I don't think Sal has done anything that I would consider to be fraudulent in the academic sense so far, as I can tell. There have just been some questionable moves at this. I am open to reassessing in either direction, however. We will continue on.
A substantial list of scientists who have done work related to human genetic deterioration include Hermann Muller, Nobel Prize winner, 1946, Cornell research professor.
All right, this is just an argument from popularity. I'm going to skip ahead a little bit, and I'm just going to say it's not the consensus of the field currently. So if you want to argue for this, you're going to have to come up with actual arguments that go beyond just this guy who is smart says I'm right, even if he does.
It's also he's conflating various different things and by citing these different people, because all of these different people were studying different concepts of genetic load in different contexts. They weren't all, and they definitely weren't studying genetic entropy. Only Sanford had any concept of genetic entropy. Everybody else were studying either mutational load or drift loads, and it's a very different thing that and I told Sal this, that he's conflating genetic entropy with genetic load, because genetic load is a legitimate field of study, and so by proxy it makes genetic entropy look better.
Right. And I also want to emphasize very much explicitly, none of these guys that were just cited would agree with Sanford or Sal on genetic entropy as being a thing. So not only is it an appeal to popularity that these smart guys agree with you, they do not.
Yeah.
Right? You can't point to, like, say, a surveyor as a flat earther and say, hey, this guy agrees with me that you can see the Sears Tower from this distance, and then say, therefore, the flat earth.
Yeah.
Does he agree with you about the flat earth? No.
Yeah. Again, like James Crow, Michael Lynch, like some of those people that he cited have said that they think that the human genome is going to deteriorate, but it's because of medical intervention, which is a whole different discussion than genetic. They're saying that we have taken ourselves out of nature, right? I think that's a silly argument. Lots of people have disagreed with them, but that's very, very different than like genetic entropy is universal and we're all going to die because of it.
Right. Savage Cobra, kind of official, for $5 says, why are we putting so much weight on geneticists that worked before the discovery of DNA? I think it's mostly before the structure of DNA was discovered. I think that DNA was discovered before these guys did their work. But I don't know, and I'm not positive that that's what we're doing, and that in part is because I don't have a great internal timeline of the history of genetics as a field. So,
yeah, I mean, I could take that from like a population genetic perspective and say that a lot of these guys actually predicted what we would find decades before we found it, and so it's kind of like in physics where you might have, you know, like a series of equations, but you don't have the data yet. You don't have the Large Hadron Collider sort of thing. And then when you finally get the data, you're like, oh, look, these guys actually knew what they were talking about. Yeah. And honestly, that's what a lot of population geneticists have done as well. As back in the 60s, we predicted that there could be no more than 20 to 30,000 protein coding genes in the human genome. And the molecular biologists were like, you're crazy. That can't possibly be right. And then it turns out that's really all it is. And they used mathematical arguments to show why. So I think some of these guys really knew what they were talking about. They're not right about everything, obviously. And like genomics has overturned some of the ideas, as you know, new data does. But we surprisingly have had a pretty robust understanding of the quantitative aspect of evolution for quite some time.
Which is really cool. And I want to emphasize how much that makes this a scientific endeavor. Right? Mm-hmm. That is, we are taking the data that we see, we are predicting future data that we might not even as individuals live to collect, and then it ends up being very close to the predictions. That is an indication that what you have is scientifically valid. I would love it if someday Sanford made a prediction about genetic entropy that then came true in an actual living population. But spoiler alerts, when it actually comes to real living populations, his predictions are bullshit.
Yeah.
Of human genetic deterioration came to the forefront because of a recent dispute between medical researchers versus evolutionary biologists,
who are not studying the same thing. One of the things I want to point out is evolution and evolutionary biology doesn't care how much you like something, right? Whereas what counts as healthy is usually defined as a desirable state regarding like the physical functioning of your body.
Yeah, he's also going to do something weird here where he is going to treat medical researchers as different from evolutionary biologists when the people that were involved in a lot of the research he's fixing to go into were evolutionary biologists. The NIH
is weird.
The NIH funds evolution. I am funded by the NIH. So he's going to go through this whole weird diatribe that medical researchers versus evolutionary biology, when that's actually not what it is. It's just biologists arguing with each other.
The dispute centered around experiments conducted by medical researchers funded by the National Institutes of Health. The National Institutes of Health, the NIH, is the most cutting-edge and well-funded medical and biology research organization on the planet.
Okay, quick pro tip to Sal. Next time you need to get an aerial shot of something, try to get something that's more than like 300 by 300 pixels. This is ridiculous. It's not an argument. There's nothing. It's just, come on, Sal. Dude, help me out here. This is painful to my eyes.
Planet. It has such a large staff, its main campus is practically a small town.
Okay.
ENCODE is one of the NIH-funded projects.
Yep.
The acronym ENCODE stands for Encyclopedia of DNA Elements, and the ENCODE mission was to investigate and catalog the function of various elements of human DNA.
I believe it was actually just to get a full genome, and then the second thing was to catalog functions and things like that. So I think the primary goal was just, let's get it so that we could have like a data file that just has the whole genome. So you can look at any chromosome, any position of the chromosome, and figure out, hey, what nucleotide is right there? And the answer will be there for you.
Yeah, the ENCODE was a follow-up to the Human Genome Project, which was like formally completed in like 2004, 2005. And this kind of followed right off the tails of it. It was like, okay, we have the genome now. Can we then go through it and predict the functions for every single, base pair in the genome? That was kind of their big science goal.
Ian Chen official says for $2 reduce, $2.99 reduce. Welcome back from the beach, Dr. Zach.
Thanks.
I don't want to shortchange how much Ian has sent me. It's $2.99 reduce. Which, in case you're uninitiated, those are Australian dollars. Because the fact that Australia did not name their currency the dollar redo is personally offensive to me.
Wait, I don't know. What is the name of the Australian dollar?
It's just the dollar.
It's just dollar? Okay.
Yeah. Whereas dollar redo was sitting right there, and they just ignored it. They were like, nope. We're going to go hang out in the Billabong spending just dollars. Like, you could have been hanging out at the Billabong spending dollar redues.
Lost opportunity.
Seriously. All right, here we go.
ENCODE and its related projects, such as Roadmap Epigenomics, 4D Nucleome, Mouse ENCODE, Psych ENCODE, etc., represent a total research investment of approximately $1 billion.
This just smacks of like a GOP politician saying, why are we spending $1 billion to find out why jellyfish are dying? And it's like, well, here's a whole list of good reasons why. And then like, let's cut it. Okay, sure, Sal.
And involved.
Okay, to be fair, Sal wasn't the one saying that. I was just saying it was reminiscent of that. Sorry, Sal. I didn't actually mean to attribute that motive or that way of thinking to you. I don't actually think that would be your position. I think you're scientifically literate enough to know why research that's important is not always intuitive in terms of why it's important.
Hundreds of medical researchers around the world, not only at the NIH main campus, but at prestigious institutions like Harvard, MIT, Stanford, Johns Hopkins, Cold Spring Harbor, etc.
Okay.
One of the accidental discoveries of the ENCODE and ENCODE-related projects is that each newborn human acquires substantial numbers of novel, albeit almost imperceptible, slight birth defects.
No, this is not true.
ENCODE
was not interested in measuring mutation rates. That's not what ENCODE was doing. And in fact, ENCODE used cell lines to do to build their like gene regulatory networks and stuff. So they were not measuring mutation rates. We knew the mutation rate the first time the mutation rate was estimated was in the 1920s. So like we've known about the idea of the mutation rate for a very long time. And that specific number has been around since I think the early 90s even. So that was not a surprise thing of ENCODE.
Yeah. Also, when he says a substantial number of birth defects, what he means is a substantial number of mutations. He is importing the idea that mutations are per se bad.
Mm-hmm.
Which
that's why he's calling them birth defects,
right,
as opposed to just saying mutations.
Now, I know it's tricky with humans, but again, remember, this is supposed to be a universal principle that just applies to organisms broadly.
Yeah.
And we have empirically measured fitness distributions in mutations in actual organisms. Am I wrong about that, or am I correct?
No, absolutely. Yeah.
And are they all deleterious?
No.
Are they all... Are none of them positive?
There are positive. There are beneficial mutations in every organism we've ever measured their fitness effects in.
Okay, so right from the get-go, Sal is importing assumptions that we know to be incorrect in order to make his argument, which means now we're going to continue on to some extent as if he had not done so for the sake of thoroughly looking at this argument. But I want to point out that right at the beginning, Sal has already lost the plot in terms of this being a viable idea in biology, right? He has already shown himself to not be serious in this particular topic. And I would extend that to some extent to John Sanford, who is making the same kinds of assumptions, although he's not phrasing it in the same way all the time. I think he's a little bit more careful in his way of phrasing these things, so it doesn't sound quite so obviously like, wait, can we really say that every newborn has about 100 birth defects? That just doesn't seem right. Like, have you ever met a newborn? Most of them don't seem to have 100 birth defects that, like, how would you define these things? Also, for $20 reduce, Anonymous Pi says the alternative to dollar redo while sitting by a billabong should have been a gym buck. Yes, that would also be a good one. I would also have accepted gym buck. Look, I don't know who it was that decided that the unit of currency for Australia should just be the dollar, but that person really disappointed. It's like when your parents used to say to you when you were a kid, I'm not angry, I'm just disappointed. That's how I feel about the name of the Australian currency.
These birth defects are called slightly deleterious mutations in the scientific literature. But unfortunately, for reasons that will be explained later, this term is misleading. A better...
All right, I'll let him wait until later, but I fail to see it any way in which this is a misleading term, but okay.
Yeah, I don't really get it either. I don't know why deleterious versus harmful. Like, they both, I mean, if we're talking about fitness effects, they mean the same thing. Like, I don't really know why it's a misleading term.
The term is slightly harmful mutations.
I don't get it, but okay.
But the important thing is that not only do babies inherit about the same number of harmful mutations their parents have, they get about 100 additional harmful mutations with each generation.
Okay, so first, no, you do not inherit all of your parents' mutations. There's a few different, very good reasons for this. One is just, you don't inherit your entire parent's genome. That's not how reproduction works. If it were, every single generation would have twice as many chromosomes as the previous one. I don't even know if I have to explain the fact that is just completely inaccurate as to how that works. So even if we just ignore...
Unless you're like a haploid. Like if you're a bacteria, but like in diploids, obviously you're only inheriting half.
Right. But on top of that, when you measure mutations that people have, generally speaking, you're getting this mutation rate from things like, like saliva samples or rubbing epithelial cells or maybe blood draws or something like that, right? Mm-hmm. What you're not doing is sampling people's gametes.
Correct.
Generally speaking, to get your genome, you're not going to the sperm or egg donation facilities to do this. For one thing, those cells don't have your whole genome, so it would be a pain. But for another thing, it's a pain anyway, especially if you are someone who produces ova, because then you need to take all sorts of drugs to induce ovulate. It's a whole big thing, right? If you ever looked into what it takes to donate eggs, I know people who have, it is not as simple as going and producing into a specimen cup.
Yeah.
So one of the things that means then is that you will catch somatic mutations that are in the body cells of someone that did not make it to the germline cells. So that's just one group of mutations that just aren't getting passed on no matter what. Further, if one of those mutations that a parent has is something that, say, is maybe lethal but recessive, if you happen to get that, and maybe just by chance as a result of, say, maybe some kind of inbreeding, not necessarily like siblings or cousins, but it could be like fourth or fifth cousins, that mutation is going to stop with you. It will not continue on. Now, it might still persist in someone else, but still, that's a mutation that occurred in your lineage that you're not going to get. So every step of the way, the number of mutations inherited at each generation is lower than the number of mutations that have occurred in the population, even in that direct lineage. So It is not true that you inherit all the mutations your parents have.
Yeah, no, definitely. Like, yeah, you're always, any kind of measurement of the mutation rate, especially if you can't measure directly gametes, it's going to be inflated because you're measuring the DNA after it's been replicated multiple times through every cell division that produced that skin type or somatic type, right? So it's always going to be more than what the germline rate is, and you have fewer germline cell divisions than most other cell in your body, right? So the germline always harbors fewer mutations by the end of your life than what any of your somatic cells harbor.
Especially, at least in humans, for females, who generally have all their gametes done by the time they finish gestation. Like, that's it. That's all you're getting. You're getting way more than you're ever going to use, but still.
In most vertebrates, not counting fish, so all tetrapods then, males are what are contributing most of the mutations.
All right, here we go.
Like the wear and tear that accumulates on a tire each time it is driven, these harmful mutations are almost imperceptible over a single generation. but over many generations, the collective effect becomes noticeable. In light of these considerations...
Oh, I almost missed the super chat. I want to say, Pillowpunk for $10 says, if every single individual of every single generation has a dozen of unique mutations, and most of those are neutral, calling them all, quote, birth defects is absurd by default. Yes, I agree. Mm-hmm. Yeah, this requires mutations to be deleterious 100% of the time to be taken seriously. And on top of that, it requires there to be no loss of mutations due to drift or selection at any point.
There's this kind of like weirdness in the way that they pitch this, that it makes it seem like the genome is just going to perpetually keep getting more and more and more and more mutant, in reality, there's an equilibrium that is reached between drift and mutation, and between selection and mutation, depending on like what's the selection coefficient against that mutation, that there will be an equilibrium value of diversity within the population. And it's not going to like just explode infinitely. It's related to the population size, because you may be adding new mutations into the population every generation, but drift is removing them every generation. And so there becomes this balance. So you're not like you're going to get just an infinitely mutant genome. It's like a pretty preposterous way of thinking about it.
And Vishanti points out, do we miss where tires are alive and passing down their worn tread to their tire babies? No, actually, we did not miss that. It was not there, interestingly enough. And Ian Chen for $2.99 Edu says, I'm going to the gym to improve my genetic entropy.
Geneticists, like Kondrashov, ask the question, why aren't we dead 100 times over? Now, the following segment is a parody dramatization of actual documented words by an evolutionary biologist.
He's fixing to get silly here.
Yeah.
Do we want to talk about Kondrashov's statement, or let him go ahead and do his rant here?
Let's cover it real quick after I read a membership chat, and then we will go ahead. Like, don't get too far into the weeds, because we'll use some of what he says as jumping off point for more detail. But David B22585, who's been a member for 9 months and is at the amniote level, says, tires avoid passing down their traits through consistent rubber use. Nice. That's clever. I like that. And by the way, I know that Embiid is in here trying to get me to make genetic load entendres. Not happening yet. Oh, wait, no, sorry. We were going to explain a bit about... So when Kondrashov said, why are we not dead 100 times over, basically, what was he talking about?
Yeah, I mean, he's pointing out a paradox. He's saying that if the theory is correct, then we should have died 100 times over. And this, we talk about this as the mutation load paradox, or in his case, the drift load paradox. That if the deleterious mutation rate is as high as we think it is, and drift is as strong as Kondrashov was assuming it was, then we should have already been dead. And so the question is rhetorical, because we're obviously not dead, right? The question is like, okay, we need to investigate why our models are not actually capturing reality. Like, why are we persisting as opposed to going extinct? So that's why it's a question as opposed to the statement, yes, we are all winding to extinction.
Right. So the solution to this is that we need a better theory, not that, well, I guess we're all going to die then.
Yeah, exactly.
Okay. Just wanted to make sure that was the conclusion to reach there. And I want to drive that home because, yes, if the paradox would actually result in the death of humans 100 times over, then there wouldn't have been a human to ask the question.
Right. Yeah.
So this is not evidence of some kind of inevitable degradation. This is why isn't there this kind of genetic degradation? Because if there were, clearly we'd be gone by now. We wouldn't have to worry about it. Okay.
Who went nuts when he was confronted with the ENCODE data. When medical researchers reported the results of the ENCODE experiments in 2012, they were greeted with some hostility. Hostility from the likes of evolutionary biologist Dan Grauer. At a presentation for the Society of Molecular Biology and Evolution 2012, as well as in official documents and in unofficial web pages on Tumblr and...
Oh, by the way, if you are in fact playing the drinking game, which I'm not, but if you are, this does count as a mention of the National Socialist German Workers' Party or prominent members thereof. Feel free to drink for that. Or if it's on your bingo card, feel free to drink for that. We will be muting most of this in an effort not to get like copyright claims from the movie Downfall, which, by the way, if you haven't seen it, it's an excellent movie. It is a dramatized chronicling of the last days of this particular individual hiding in his bunker, realizing that his entire empire is crumbling around him. Which, yeah, I think was well acted. I don't speak German, but still, it was very... it was a worthwhile watch, I'll say that. I recommend Downfall. It is not sympathetic to the character on screen. So, yeah, there's that.
His university website, Grauer declared...
Okay. Now, he did say that. He did say, that is a direct quote, if ENCODE is right, evolution is wrong. What did he mean by that? Because I feel like we get that quotation a lot from the young Earth creationist crowd, but it's very rarely in context, nor are Dan Grauer's thoughts on that ever really explained. So
yeah. So I think, for one, we need to understand who Dan Grauer is. Dan Grauer is a very well-known molecular biologist and evolutionary biologist. He's written multiple textbooks. He's just like, and he's also like sort of a fiery character. He has a very big personality. I met him at the university. He's from the University of Houston, and he came to where I did grad school and gave a talk. And so I got a chance to meet him. He's just a very, very fiery person, as you can see from like some of the quotes that Sal is using here.
Yeah, we're playing through this because it's not adding substantively to the argument. It's just a little bit of a meme. And to be fair, I do like this meme. I've seen numerous versions of it that I enjoy. So I'm not going to fault the meme, but it's not adding anything. So that's why we're playing it as we go.
Yeah. What he meant in that specific case, that if ENCODE is right, evolution is wrong, I think is actually a very important quote. Because what he's trying to capture here is that we have a quantitative understanding of the relationship between things like selection and genetic drift. And we have had this understanding for 100 years at this point. And humans have historically had small population sizes, which means that drift should be pretty dominant in humans, which means that it's very, very unlikely that selection has somehow, one, created a perfect genome that is entirely functional, and two, that a genome can be maintained in the absence of selection. Because that's one of the things that ENCODE found, right, is they claim that 80% of the human genome was functional. However, only about 10% of it show any evidence of selective constraint, any evidence that mutations are being actively weeded out by selection. And so what Grauer was pointing out here is that it seems like a genome can just persist forever, like, and no mutation can ever harm it. And if that's true, it's hard to see how evolution could be correct. But it's very difficult to see how that could be true, right? That the genome could be maintained to be 100% functional and never, ever, over 80% of it, have a single deleterious mutation occur.
By the way, Inkboy, who's been a member for 14 months, that's quite a while, and is at the amnio level, says, talking about someone and having negative visuals is common propaganda tactic to play on viewers' emotion, which is odd because Sal actually wants us to agree with his version of Dan Grauer here. So I don't actually think that's what he's doing, because for Sal, in the narrative of this video, Grauer's the good guy. But one of the things I want to point out is that his use of that, if ENCODE is right, evolution is wrong, is almost directly opposite to what it's actually stating, right? This is used as, well, since we know that ENCODE was right about at least 80% functionality, I guess evolution is wrong. Time to go back to the drawing board. But one of the things I want to point out is that was based on their 2012 findings, right? That roughly 80% functionality.
Correct.
To my understanding, 2 years later, that number was walked back by ENCODE.
That number was very strongly walked back. So why this meme doesn't really work for this situation is because Grauer won this argument. Grauer and the many, many, many biologists that fought against ENCODE right after that 2012 publication, it was a firestorm. It was a big deal. They won the argument. They won the day. ENCODE rolled back their initial statement. And so it's curious to me that so many creationists have continue to cite the original ENCODE study, even after ENCODE themselves have already taken the L. Like they recognize that original statement of 80% was misleading to the public. And in subsequent publications, the lead authors rolled it all the way back to 20%, even though I would argue that even the 20% has very little empirical support for. So yeah, ENCODE itself won't even stand behind those numbers.
Okay, so now recently, while working on a different video, I checked with a list of indications of pseudoscience because I wanted to compare certain ideas to things that seem like pseudoscience. Now, one of the big things that they said was failure to take into account data that contradicts, but accepting conclusions that at least superficially seem like they agree with the pseudoscientist. Are you telling me that maybe people arguing for genetic entropy might be acting like pseudoscientists?
I mean, that would seem like the correct conclusion to come to.
My goodness. Now, this kind of came about from John Sanford, who we all know has the galaxy Chad brain that the likes of which the human race has never seen before, right? This is clearly the smartest individual ever to have lived. But he's ignoring data, apparently, as far as we can tell, that would actually undermine his conclusions. So he probably isn't just honestly mistaken about this. So when I called him dishonest for this kind of thing, I don't know, it seems like it's fairly reasonable, unless someone wants to argue with me that the inventor of the gene gun, who's in the Smithsonian Institute not once but twice, unless you want to argue that this man is in fact just a moron, it seems hard for me to come to other conclusions. Just wanted to put that out there since, this is a response to a set of videos that the responder didn't bother to watch. Which, I'll point out, hey, what am I doing right now? I'm watching one of the responses so that I can respond to it.
Call medical researchers at the NIH ENCODE project crooks and ignoramuses. Why did Grauer call the chief architect of ENCODE, Ewan Burney, the scientific equivalent of Saddam Hussein?
I don't know. That's probably not something I would have done.
Yeah, he did say that. Grauer, again, like I said, Grauer is an over-the-top character.
Yeah, look, I'm not here to defend everything that Grauer has ever done either. That's until you attempt genocide via poison gas, I don't think you should be compared to Saddam Hussein, generally speaking. That's where I am with it. But let's talk real quick. What was the definition of functionality that ENCODE used that got them such a high number? Because
I think maybe we should even point out the fact that Sal has not actually engaged with Grauer's critique. Sal has sort of led into this whole little meme by talking about the mutation rate, which has nothing to do with Grauer's critique of ENCODE. Grauer's critique of ENCODE was multifaceted, but one of his main critiques was the definition of function that ENCODE used. And you can think about biological function as being defined in one of two ways. We can either define it by the causal definition, that is to say A causes B, therefore the function of A is to do B, right? It's just a very simple A leads to B causal definition of function. This could be something like this region of the genome binds a transcription factor. What's the function of that region? Binding transcription factors, right? That's the causal definition. Mm-hmm. Now, that particular definition is problematic in biology because things do lots of stuff that's not necessary. So what we tend to go with is what's called the selected effect definition. And Grauer uses a really good example, and I think it maybe can drive home the differences between these two ideas. So if we think about what's the function of a shoe, right? If the function of a shoe is anything that a shoe does, which would be the causal definition, that if I'm walking down the sidewalk and I step on some gum that binds to my shoe, the function of my shoe is to bind gum, because that's a thing that a shoe can do, right? Mm-hmm. However, we would be hard-pressed to think that a shoe was designed to do that we actually wear shoes because we want to pick up gum from the sidewalk with them, right? What we actually wear shoes for is to protect our feet. And so you can imagine the case in which the shoe becomes so tattered that it fails to protect your feet, but it still manages to bind gum. you would say that that shoe is not functional. And so we distinguish these two things based on why has this particular region been maintained. So does it bind a transcription factor by chance? It just happens to bind one, but there's no transcription that ever occurs there. There's no start codon within a megabase, distance from that promoter or from that transcription factor binding site. Like, we need to do more than that. Is that transcription factor region, that transcription factor binding region, preserved by natural selection? That is to say, every individual you see has it, and when it binds that transcription factor, it transcribes a gene, right? So there's two different things. One would allow you to basically assign function to anything that has an action. Any kind of biochemical action in the genome will get given a function. Well, it binds a transcription factor. That must be its function. Whereas the selected effect definition will discard the frivolous things that happen, because the genome it's a chemical, it's a biological molecule that does lots of frivolous things. And so we want to be able to distinguish between those things that what we think of as functional, that is to say, like we were referring to the vitamin C, that's necessary to be there, versus those things that aren't necessary.
Now we got a couple super chats I want to read real quick. Pillpunk for $5 says genetic entropy because everything was better back in the good old days. Yeah, it does kind of smack of that. I have a history degree, and one of the things you can see going back in history is that basically a constant refrain from basically everyone who's kind of old is that, the damn kids these days, they're screwing up their language, they don't have any more respect, it was better when I was a kid. And it's like, yeah, well, your parents were saying the exact same thing, buddy. It's been happening for millennia.
Oh, yeah.
Savage Cobra Kinda Official for $2 says the function of a shoe is to be thrown at bush. Yes, that is in fact a valid function for a shoe. That might be the best function for a shoe.
Agreed.
I still have vivid memories of watching that on the news, and yeah, that was it was great. Anyway, let's again go back to this, keeping in mind that at the risk of sounding tone policey after having said that, the only substantive response I got was tone police, I still don't think it's really appropriate to compare people to murderous dictators who are not themselves murderous dictators. So,
no, sure, for sure.
Why does Grauer use a picture of toilet paper made of $100 bills to describe the NIH's investment in ENCODE?
He thinks it was dumb.
It's because he's mad.
I mean, I think my explanation was a little bit more specific, but yeah, also.
The question is, why is he mad?
Why you have to be mad?
Grauer is mad because he's an evolutionary biologist, and it was...
Saying it that way sounds to me like, why did the plane crash? And your answer is gravity.
Yeah.
Kind of, but there might be something more specific you could point to.
I love the way he's reading Grauer's name. Like, he's just getting like progressively growly about reading Grauer's name.
Yeah, well, I mean, it sounds like growl. You might as well growl when you say it, right? Yeah, he does that throughout the whole video, and it's a little odd, but like whatever, it's fine.
Grauer himself who growled, "If ENCODE is right, evolution is wrong."
Yeah. That's why I prefer. Oops. Don't want to get a copyright claim.
But to Dan Grauer's credit, on his official university website, he shows a cute picture of his granddaughter, albeit he puts the caption, quote, "My granddaughter, Lila Cachette Grauer, gives ENCODE the finger," end quote.
Okay.
But all ENCODE did was generate data. It's only data. So
I want to point out that this is mostly Sal's like weird attempt to like tone police Grauer and not really engage with Grauer's argument.
Yeah.
Because Sal is just accepting the 2012 ENCODE numbers without any recognition that even ENCODE isn't standing by them. And that while perhaps their walkback was not as much as Grauer would have liked, it was a whole heck of a lot more than what Sal would want them to walk back.
Yeah, and I mean, he could have pointed out, like, if he didn't like Grauer's tone, and many people didn't, you could have looked at, like, W4 Doolittle's. You could have looked at Larry Moran. You could have looked at Alex Palazzo. Like, a lot of these other biochemists and molecular biologists were also very critical of ENCODE. They just didn't get as much publicity because they weren't as loud about it as Grauer was, right? Like,
Also didn't give the easily quote mineable quotations. So you got to realize that by and large, when creationists are looking for sources, they're looking for sources that they can most easily twist to agree with them, even though they don't.
Yeah.
Because, I mean, let's be honest, any source that agrees with the young Earth creationist is going to be pretty trivial in terms of its scientific work.
Why does Grauer have to be so mad?
It's only a game. Why you have to be mad?
So why did Grauer say, if ENCODE is right, evolution is wrong? To understand why, first, let's do a mini history review. Edward Blyth pioneered the idea of natural selection. Then Charles Darwin.
Okay, at this point, he's just like, Charles Darwin didn't come up with natural selection, so therefore it's, like, I don't know. I don't know what the point of, like, I don't know, understand the rhetorical point of this whole thing. Because the history of science doesn't have a whole lot to do with what's going on in science right now. So, like, it doesn't matter who came up with natural selection. It doesn't matter who came up with survival of the fittest. It doesn't matter who came up with the word gene or mutation. None of that actually matters to the current ongoing research. Right? If everyone just acknowledges that Blyth came up with natural selection, that doesn't change the current research. And one way in which...
It also misunderstands, like, the purpose of precedent in science and how we understand precedent. Because even if somebody stumbles upon an idea, if they don't understand what they stumbled upon and use it in the context that we currently use it, like Darwin and Wallace did, then they don't get the credit for it. And I can give a very specific example. So, like, the Hardy-Weinberg principle was discovered in 1908 by G.H. Hardy and Wilhelm Weinberg, but Carl Pearson and Udny Yule wrote about it 10 years prior. They stumbled on the same thing, but they didn't understand what they had stumbled on, right? And so we don't call it the Pearson-Yule principle. We call it Hardy-Weinberg because those were the people that actually recognized what it was. And so it's not merely that you stumble on a thing. You have to actually promote it and understand the importance of the discovery that you made to be given credit for it.
Yeah. I do want to point out that is also not to say that the people that we attribute to things are not sometimes the wrong people, and we should not give attribution to other people who actually did do the work before them. One of the more famous examples that I'm most familiar with, Mary Anning, who was a key player in the early days of paleontology and whose contributions were ignored for over a century. Now she is widely acknowledged for her prowess at fossil hunting, at describing fossils. She was constantly being asked to consult on finds or papers or things. And that's important. But I also want to point out that when you started really recognizing her contributions to the field of paleontology, it didn't change the science of paleontology. No one had to retract a paper because their conclusions were wrong, because they misattributed something to some other scientist instead of Mary Anning, right? Like, that's not how it worked. I want to point that out there, too. All right. Here we go.
That evolution will lead inevitably to progress of lesser life forms toward greater life forms. But Spencer and Darwin's ideas have flaws that should have been brutally obvious from the start, but which have been mostly ignored until recently.
Oh, this will be interesting.
Spencer and Darwin's theories fail when something I call Muller's limit is violated.
All right, let's talk about what Muller's limit is in this context so that we have a good idea as to what we're talking about going forward. So first, Muller is primarily concerned with, I mean, let me know if I'm wrong, because this is not my area of expertise or anything. He's primarily concerned with the effects of genetic change in primarily haploid organisms that do not have any significant crossing over events or horizontal gene transfer. So basically, they're not getting genetic material from outside a direct ancestor-descendant clonal type relationship, where the only genetic variation comes in through mutations during these clonal events, right?
Yeah, that's called Muller's ratchet, and that's where that specific idea comes from. That if you're a haploid, you can't recombine out deleterious variation. Now, what he's fixing to talk about in terms of Muller's limit actually comes from the 1950 paper "Our Load of Mutations." Well, it actually comes from Haldane 37. Muller was just kind of citing it, but regardless, it's more about the idea of just mutational load in general. So, what's the proportion of deleterious mutations that any population is going to harbor, given some deleterious mutation rate and a mutation selection equilibrium?
Okay. Now, Eric311, who's been a member for 4 months and is at the amniote level, says, it's funny that Darwin is to young Earth creationism what Neil deGrasse Tyson is to flat earthers. He's their science bogeyman. Yeah. I mean, he does seem to be, right? Like, they both cite this one guy as if you disagreeing with him means that you've left the fold, and as if they're, like, the origin of all the evils of the scientific world. It's a little weird. Because one of the things that... I have a satirical set about this. I have a satirical Darwin temple set. Specifically because of the number of times that creationists like to accuse people of treating Darwin as if he's some kind of prophet or, like, God or something. And it's like, no, he's just a guy. He got a lot of stuff wrong. It doesn't really matter that he got stuff wrong because we've moved on. We moved on a long time ago. Yeah. They're projecting that. Like,
this is the way they view the world, is that they have prophets and they have faith-based systems and they have religions. And so they just assume that we see, people in the exact same way. So anybody that we might look up to scientifically, we must see as a prophet that can do no wrong.
Yeah.
And that's just a silly way of thinking about it.
It's also not even a very traditional way to think about prophets. Most religions who have prophets don't think that their prophets are infallible.
Yeah.
Which is, like, yeah, sure, Christians and Jesus, but, like, look at the biblical depiction of Moses, who's probably the most famous prophet in the Bible, if you're going to take out, the Christian sections of the Bible. Dude screwed up a whole bunch.
Yeah, I want to also say here that the critique of Darwin and Spencer's view of natural selection is incredibly outdated. What we should be talking about natural selection as understood in a more modern context, and that was by the work of Fisher, Wright, and Haldane. Darwin's view of natural selection, and especially like survival of the fittest, has long been recognized as a tautology. It's not a very useful way of thinking. Like, obviously the fittest will survive. Like, that's not a useful way of thinking about it. For natural selection to be at work, it's not about just who is the fittest. It's about, are there genetic variants contributing to that fitness, right? Because you can be fit just by the environment, you can be fit by quirks of development, but that doesn't get passed on. For there to be an evolutionary response, for natural selection to actually drive something, it needs to be heritable fitness contributions, right? And so, like, that's why survival of the fittest is not a very good analogy for the way we actually think about natural selection.
I'm going to play a bit more. I think we're actually getting through this at a decent pace, but in a little bit, we're going to take a quick break to do things like refill drinks or anything, and then we will be right back at it. So we're going to get a little bit more, talk a little bit more, and then probably take a quick break.
Call it Muller's limit, after Hermann Muller, who won the 1946 Nobel Prize for his study of mutations caused by X-rays. Muller was among the first, if not the first, to compute this infamous limit. He published the computation of the limit in his 1950 paper entitled...
I just want to point out, this is once again, we're confusing mutational load with genetic entropy when those are not compatible ideas. They're thematically similar, I suppose, but that's not enough to say that they're the same thing.
Yeah.
Titled Our Load of Mutations. His paper also overturned the prevailing view of the effect of mutations on the human genome. Muller was very disturbed by the potential damage that X-ray radiation and other man-made radiation could cause to future generations of humans. He wanted to know the limit of radiation-induced mutations, which the human genome could tolerate and still survive long-term as a species. This limit is what I call Muller's limit. Whether intentionally or not, this limit also defines the critical conditions under which natural selection and survival of the fittest would fail. It would fail whether or not the cause of mutations was man-made radiation or from pre-existing natural causes such as natural errors in DNA replication. When Muller's limit is violated,
it would be great if we had a definition of Muller's limit at this point, right? Because we've been talking a lot about dangers of reaching Muller's limit. Okay, what is it, Sal? Back up a little bit.
Yeah, I think he's going to give it in like just a second, but
Okay.
he doesn't. He doesn't make it very clear. I can explain it once he gives it.
Okay, yeah, this isn't so much like a point of fact or science thing. This is like a how to structure argument. Before you
Yeah,
want to start talking about the results of a thing, you want to make sure everyone knows what the thing is.
yeah.
Natural selection and survival of the fittest will fail. Another way to think of it in a more lighthearted fashion is Darwin goes bonkers when Muller's limit is violated.
I'm sad that Darwin didn't hit the corner.
Muller's limit for humans is approximately one harmful mutation per individual per generation. The meaning of this will hopefully become clear by the end of the video.
Okay, so what Sal is going for here is that I want to try that this is me trying to steelman him. I want to try to get as good a handle on what he's saying right now. Saying that if more than one harmful mutation, so deleterious mutation, occurs per individual per generation, that will tend to mean that the human species is on the road to extinction. Is that
That seems to be what he's suggesting. He's portraying this in a little bit of an unclear way. So it's not strictly one harmful mutation. It's whether that it's like the proportion of mutations that can occur in regions of the genome that are even capable of having deleterious mutations. So if we say, what's the functional fraction of the genome multiplied by the number of deleterious mutations that you think is going to have as a proportion of that. So then that gives you basically what we would call U. And the equation is like L equals 1 minus E raised to the negative U. And the negative U is this value. So if you plug in 1 to that value, what you basically get is like 40% of the population fails to reproduce if you plug in 1 for that value. What that means is that females need to have, on average, around 3 offspring. And if they have around 3 offspring on average, then that's a mutational load that humans can tolerate.
now historically that's well within the
Exactly. Yeah.
reproductive rate of humans.
Yeah. And that's why he's calling it Muller's limit. Is he's saying anything greater than 1, because the shape of this curve is like exponential, and then it reaches 1 really quickly around 1.5 to 2. I mean, it gets there really, really fast. And so basically anything above 1, the proportion of individuals that fail to reproduce becomes so high that the human reproductive output couldn't keep up with it. If we were fruit flies, we could keep up with it, right? If we were organisms that had tons and tons of offspring, we could keep up with it. But mammals, especially mammals, big vertebrates, they can't keep up with it because they don't have that many offspring.
Right. Actually, I did some quick fruit fly math the other day. A creationist was saying that we could take the human population growth rate and just recess it back smoothly using a decay function into the past to find that humans started with just 2 people about 7,000 years ago. And I said, okay, well, let's do that forward, but with fruit flies. And that will result in approximately a number of fruit flies roughly equal to 10 to the 82,839th power. Which,
Jeez.
I don't have to tell you, does not match the world around us, especially given that the number of electrons in the observable universe is estimated to be about 1 to the 80th.
Yeah.
And I don't know if you guys know this, but as it turns out, most fruit flies are composed of more than 1 electron rather than a tiny fraction of an electron, which isn't possible, by the way. So just tossing that out there, because I like to give creationists like a free order of magnitude because they're usually off by 6 or 7, but in this case, wow, one order of magnitude is not going to be nearly enough to fix that problem.
Yeah.
In the meantime, as a shorthand, I'll simply say Muller's limit is approximately 1. The reason evolutionary biologists were upset at ENCODE is that ENCODE experiments suggest that the human genome is experiencing a rate of mutations on the order of 50 to 100 times beyond Muller's limit.
Now, in this, he is assuming, because we talked about earlier how this is dependent on the functional fraction, right? So he is assuming here that the 80% is roughly accurate in terms of what actually the functional fraction of the human genome is. Is that what he's doing?
Yeah, so he's saying 50 to 100 is the average number of mutations that any individual has. ENCODE did not find that. That is not ENCODE data. ENCODE, again, we're using cell lines. They were not measuring parent-offspring mutation rates. They were just looking at cell lines. Some of them were even HeLa cells. Some of them were cancer cells. I mean, there's many reasons why Grauer was critiquing ENCODE, and one of them was the cell lines that they were using, because many of these cell lines are transcriptionally very permissive. That's just kind of an aside, but I want to make clear that ENCODE did not come up with this number. And so that was not part of Grauer's critique. But yeah, the 50 to 100 is just based on the pedigree mutation rate that has been estimated in humans. But that says nothing about how many of those are deleterious. Okay. And it says nothing about where they occur. It's just like that's the approximate number you expect on average individual should have.
So what I want to see if my interpretation of what Sal's trying to do here is something that you agree with. That's what he's going to do, or he's trying to do. Then we're going to take a quick break. We're going to refill our drinks, and we're going to come back because it's hot and I need some ice water. So for this to make sense, right? We'll ignore for now that it's not ENCODE that produced these mutation rates. It's other groups, but this is, I believe, a reasonable estimate for the mutational rate for humans, right? Or the pedigree mutation rate.
Yeah.
Okay. So in order for this to make sense, one, we have to assume that the functional fraction of the human genome is very high.
Near 100% to make these numbers work. Yeah.
Okay. And we have to assume that all mutations are deleterious.
Yeah.
Now,
exactly.
we know empirically that neither of those things are true.
Yeah.
Okay.
Yeah.
So I just want to make sure that I understood, because the thing is, look, this isn't my area of expertise. There's a reason I'm having you on, because I want to make sure that my understanding of what Sal's argument is actually what it is, because I don't want to strawman Sal, because,
sure.
What's the point of responding to an argument when you don't understand the argument, whether because you simply fail to understand it or because you didn't watch it?
Yeah, and all of these ideas are mutually like exclusive, because if 100% of the genome is functional and any one of these, then any base pair can mutate to a less fit variant, then you should see selection acting across the whole genome. The whole genome should be selectively constrained. And we see that in things like bacteria, right? That have a very, very high functional fraction of their genome. We do not see that in humans. Only 10% appear capable of having a deleterious mutation. And so like his numbers just can't be right. It would be, again, you would need 100% of the genome to be functional, and you would need to see selection acting over that whole thing. And that's just not what we see at all.
Now, Nestle20 asked a question, but I'm not sure I understand it. So what I'm going to do is we're going to take a quick break. And Nestle20, if you would like to rephrase that, I think I feel like you're missing like a clause or something in there. Maybe a verb, I'm not sure. But we're going to come right back after just a quick break to refill drinks. I'm also going to play some ads because I like having this thing called food and this thing called a place to live, and that helps with that. So I hope to see you all here on the other side of the break, and we will be right out of here. Hey everybody, we are back. Before we get right back into it, I just want to say a quick few things. One is, thank you so much for helping me get to 10,000 subscribers. I'll be honest, if you told me 3 years ago when I really first started consistently uploading to this channel that I would be at 10,000, like 10,000 crazy people would want to hear me ramble about like fitness effects and functional fraction and things like that, no, there's no way that's ever going to happen. And yet here we are. I have no idea what's wrong with all of you guys that you think that this is entertainment, but hey, I'm not going to complain about it. That being said, the fact that the channel can so consistently get 2 live streams and a scripted video out week after week, with only the occasional swapping out of a Saturday stream for a Saturday upload, it's entirely down to the fact that my fans are very generous and are supporting the channel, whether it be through super chats or what I'm going to tell you about now, which is various monthly ways that you can pledge some small amount of money. There's a Patreon. There's also the channel memberships. Channel membership, there's a join link right below. The Patreon is linked in the description. Also want to say Bent Hoven, my frequent co-host, has a Patreon. If you like his music, you can get behind-the-scenes access to some of his music. And that is either monthly or annual. You get a 12% discount for going annual. And it really, really does help. It is a resource stream that does not depend directly on things like the algorithm. Because one of the things you might know is that my most recent series just did not get picked up in that algorithm. The algorithm hated it, and I don't know why. It had good like click-through numbers on its like how many impressions it got versus how many clicks. Just didn't get very many impressions, which is just down to the algorithm. So that can help with that. You can get all sorts of perks. There's about 5 months worth of scripted videos that are just waiting for you. Sign up to either of those. You can just spend like the entire rest of the day doing nothing but watching Dapper Dino videos for hours. In fact, actually, I'm hoping today to get another video uploaded, which will be the third part of my quick facts response. ICR put out these quick facts. But also, real quick, if you're not comfortable, you can't afford it, or you just don't want to do a monthly or an annual thing, there are a lot of other options. I have a merch store in the description. I also have an Amazon wish list, as does Bent, also in the description. So if you just want to get us a little something, that's in there too. And lastly, I just want to say I'm hoping to have a 10,000 subscriber special a week from today, in case you missed that. And then we have a few things to read. Piltbunk for $5 says, I've yet to see a young Earth creationist explain how healthy, functional baby humans continue to be born with genetic entropy is real. Yeah, that is curious. One of the things I think we'll touch on is, if this were a real thing, this genetic entropy, the human population would be declining. It's literally impossible for genetic entropy to work and to have a growing population. It's like logically contradictory. We also have, for 20 pounds, almost said dollars, unverified real B2022 officially not official. Goodness. Says, just sending this to support the channel, my good Ceratosaurus, with a dapper and a tunicate, which, oh man, that tunicate. I don't know why you guys do it. And you're doing it again. I know it's because of gifted subscription. Send tetrapod emojis, guys, not the pelagic tunicates. Those are gross. Scare people away. Maya Atkinson sends a $5 super sticker, which is, I believe, a tiny lime and a larger lemon high-fiving. Thank you very much. Heather Kuhn for $2 says, apparently DNA was first isolated in 1869. That actually sounds about right as far as just getting the chemical. It's actually not very hard to separate out DNA. Like, you can do it at home if you have, like, some soap and a blender. Now, can you sequence that at home? No, probably not, but you can look at it. Let's see. Now, Savage Cobra Kinda Official has gifted 10 Dapper Dinosaur memberships. Thank you very much. That's extremely generous. And I want to read out the names that went to. So that went to Crypto457, Bio Alley, Rhea Jays, or Raya Jays, I'm not really sure. Please forgive me. Charles Starbuck, Wayne Gaffney, Christopher Kennedy Atheist, Eric Davis, Morena, Dr. Legendre, I think, RD, and it looks like that is it. Welcome, guys. If you want to get access to the special Discord server that is just for members and patrons, you want to go to Discord, go to Settings, go to Connections, and add your YouTube channel. The wrong way is to send me a DM asking me how to do it, or ask me for the join link. Those are the wrong ways to do it. That's the right way. We also have $2 from Strawberry Vein, who says, people like you, Dapper, deal with it. Okay, fair enough. We also have Lance Hall, who says, for $5, sliding in late. That's perfectly fine. Nestor20 says, my question was about the proportion of people who fail to reproduce regarding mutation load. Is that these individuals don't have any kids at all? And I believe the answer to that is yes. If this mutational load were like 40%, that would be 40% of, and I believe it's specifically females, right? We're counting children per female.
Yeah.
So 40% of females would fail to reproduce. Whereas if I remember correctly, it's actually like a large majority of human females actually do in fact reproduce, much higher than the proportion of males who do.
Yeah.
Okay. All right. Well, I think with that, unless you have anything real quick you want to add, we're just going to dive right back into this.
Let's do it.
Here we go.
So what does this mean?
Houston, we have a problem.
Say again, please.
Houston, we have a problem.
Hey, we've got a problem here.
Whoa.
Look, much as I appreciate this, it's also not actually true. We would have a problem if the basics of this were true. Steve Reno for $2 says, let the pelagic tuna get into your heart, Dappa. No, absolutely not. Then they would just like swim around inside my bloodstream. That's probably fatal. I don't want a pelagic tunicate in my heart. I don't even want a sessile tunicate in my heart. I like those guys. Dan, here's a cool thing, Zach. Zach, did you know that tunicates have two-way hearts? They actually spend part of their time pumping blood one way, then their heart switches direction, and they spend about the same amount of time pumping it the other way. And it's about 10 minutes.
I did not know that. I know they have a really weird feeding apparatus, the gill basket.
Yeah, the gill basket.
I've dissected one of these before. They're really, really strange critters, but I didn't know that about the heart.
Yeah. There's a lot of really cool stuff you can learn about tunicates. You just don't have to go into like the free swimming ones. You can just study the ones that stay put on rocks.
They're easier to collect anyways.
Exactly. They're also prettier. Like the free floating ones tend to just be transparent blobs of nothing, whereas the ones that stay on the rocks can have like cool patterns and colors and also, you know their mantle is made of cellulose?
I didn't know that.
Yeah.
It's like rubbery. I don't know if you've ever held one, but they're very like kind of rubbery and
that outer layer is
squirt at you.
Yeah.
The common name is sea squirt, and when you pick them up out of the water, they will squirt at you.
Yes, they will. But yeah, their little exoskeleton-like shell is it's made of cellulose, which is weird. It's the only animal I know of that makes cellulose and uses it for. Yeah. Lockulators for €5 says just because sunglasses thumbs up. Well, sunglasses thumbs up to you too. Although I'm inside, so I'm not putting on sunglasses. But maybe later. All right, here we go.
Hey.
Some will say ENCODE numbers are inflated. So let's go back to pre-ENCODE estimates.
Okay, I want to point out some will say that ENCODE numbers are inflated, like ENCODE. Right? That's some of the people who say that those 2012 numbers are inflated, Sal. I'm assuming that Sal is either watching or going to be watching. I know he was... I could tag him earlier in chat, so I think he might still be here. Yes, Sal, the people who think that this is inflated is literally ENCODE, among others. So when you say some people, maybe in the future you should say some people, including ENCODE, who no longer stands by the numbers I like. Maybe say that, because that would be the honest thing to say. That would be the well-informed thing to say. And if you don't, sure, you're either you're either not well-informed or you're not being honest. But you should strive to be both. The unfortunate problem is that when you do both, you can't also maintain being a young Earth creationist for very long. Now, unverified real B2022, officially not official, has gifted 5 Dapper Dinosaur memberships, which went to Saw B, The Fallen, Peter Flemberg, a Coyote, Lord Crocosquirrel, and I believe that is... Yep, that's 5. So thank you very much for your generosity. All right, here we go.
Of 10 for the number of harmful mutations per individual per generation. That's still 10 times beyond Muller's limit. And in that case, Darwin still goes bonkers. And that means...
Houston, we have a problem.
Say again, please.
Okay, Sal, in the future, don't repeat this exact same meme. You want to try to get like a quick other meme that has the same kind of theme.
I present...
So like, pick a different movie where something really bad is about to happen and people like announce it. Like maybe you want to go with like the classic bit of Star Wars where, you know, Han Solo says like, oh, I've got a bad feeling about this. Like, do one of those. And this is just like how to structure your videos and kind of thing. A couple things. Pelagic Tunicate Truth Official for €2 says, don't forget that AIG is a bag of dicks. No, do not forget that. Zach, I don't know if you've seen any of the drinking streams, but we have some drink triggers, and one of them is that anyone who super chats anything to that effect, we take a drink, and that's because AIG has a notorious history with falsely copyright claiming other people's videos when they use fair use clips of their stuff. In fact, I once had a video copyright claimed by AIG, in which one of the clips that I showed was AIG themselves using a Hollywood movie under fair use, which. I just love the delicious hypocrisy in that one.
Yeah.
And for $5, Starberry Vane says, so what you're telling me is that tunicates were specially designed. That is not, in fact, what I'm telling you. But they are very cool and very weird among the chordates. There's really no chordates that are anything like tunicates. Despite the fact that they're actually more closely related to vertebrates than like lancelets are, even though morphologically lancelets are much more like juvenile chordates than tunicates are. It's a weird thing. All right, here we go.
And here a purely pedagogical/educational model, which encapsulates the essence of the problem posed by Muller's limit. Even though this pedagogical model is inaccurate, the problem of Muller's limit remains, even with more biologically realistic models of human evolution. Again, what you're about to see is a purely pedagogical model. It needs to be refined to be biologically realistic. But the essentials of the problem posed by Muller's limit should be apparent through this model. This pedagogical model is a females-only model, where there are no dads nor sons, only mothers and daughters.
Okay.
Because this is a females-only model, in this model, girls rule the world.
Okay, well, first we're going to mute so we don't get a copyright claim there, but also, why not just say that this is an asexual population? Because that's just what he's saying.
Yeah, I think he's also fixing to present a model that has nothing to do with Muller's limit. So maybe we should talk a little bit about like what Muller's limit actually is like the problem that arises from it and the problem that arises from human mutation rates before he gets into this kind of weird thing that he's fixing to do.
Let's do that real quick after I read a couple chats here. So Fade to Gray, Untruth, Semi-Official. Who's been a member for 12 months and is at the amulet level, says AIG equals bag of dicks. ICR equals clown. Dinosaur Adventureland equals skull and crossbones. Yeah, there's already been at least one fatality on Dinosaur Adventure. And then, of course, Dapper plus Pelagic Tunicate equals yin yang. That last part I have to strongly disagree with. You guys trying to get me to like Pelagic Tunicates. It's not going to happen. Unverified Real B2022, officially not official, for £5 says AIG is a noxious network of knobs, numpties, and nincompoops. Which, yes, that is true. All right, sorry, Zach, go ahead.
Yeah, no, I was just going to say that. So he starts off by saying, by basically proposing that 50 to 100 new mutations per generation are deleterious, and he calls that the ENCODE data. And then he says, even if we go back to the pre-ENCODE data, the deleterious mutation rate was 10. That's about the highest I've ever seen it estimated, but that is from empirical studies of the deleterious mutation rate as high as 10. And in these studies, they discuss what is called the mutation load paradox. Because if you, as I talked about before, if you plug in 10 into the equation of the mutation load, the 1 minus e raised to the negative u, what you effectively get is that 99% of the population don't survive to reproduce. That's not at some future date. That's not like, oh, we're going to degrade. That is tomorrow, then the next generation, everyone's dead, right? So that's the mutation load paradox. And that's why when people say, oh, we found that the deleterious mutation rate was 10, there is something else we're missing. There's something about the mutation load mathematical models that are not accounting for how each one of the independent deleterious mutations are interacting with each other. Because what these models are assuming is that they interact additively, that you can just like add the selection coefficient from each and every one of them up, and then that sum represents the cost of the population. But it's very possible that's like a super simplistic way of thinking about it, but it's very possible that these mutations are interacting epistatically, that they have various different, so like if you have more than 3, for example, then your fitness goes to 0, right? So like there's many different ways in which these independent deleterious mutations may not be independent. They may be interacting epistatically that would completely nullify this idea. And so when we talk about solutions to the mutation load paradox, or talk about how can the Muller's limit be violated and humans still persist, what we need to think about is why is basically that our simplistic models are failing to capture the complexity of reality, and we need to fix the models. Not that we need to like save humans. Humans are going to be fine. It's the models that are currently failing.
Right. And I want to drive home, like you said, this would mean that 90% of females currently in the present would fail to reproduce.
99.999, like the vast majority of females would fail to reproduce immediately. The population would collapse in a generation.
So one of the things that I want to point out there is that would, if we just go with 90%, right? Mm-hmm. We'll be very generous. That would mean that if you took all human females currently on Earth and tracked all of them, there's about 4 billion of them, roughly, tracked all of them till the end of their lives, only about, what is it? Yeah, like 360 million of them would actually end up being able to reproduce. Mm. I guarantee you that of the currently living human females, way more than 360 million of them will manage to reproduce. So we know that this model cannot be right, because the important thing in science is that your model reflect reality as we find it. Yep. If your model does not work out in the real world, it might be a fine bit of math, but it's not scientifically useful. Go talk to a mathematician who cares less about the real world. That's fine. But when it comes to real life science, no.
Yeah, and it's even worse than that, right? So like even if the 360 million do reproduce, to maintain a constant population size, they have to make up for all of those that didn't, right? So they have to have themselves hundreds of offspring to make up for it. Right. And like the human population size has been growing exponentially for quite some time. So like if the mutation load were really a problem, our population, one, would already be dead, but two, it absolutely couldn't be displaying population growth. Right? Those two things are fundamentally in conflict with each other. So
this isn't just a theoretical thing. We currently know that reality is at odds with the proposed model. So the proposed model is wrong.
Yeah, that's why it's a paradox. If we wouldn't say it's the mutation load paradox if it was real, we would just say it's the mutation load, right?
Right. Okay. Well, I'm going to hit play, and hopefully we'll get past the sound bits, because YouTube loves to just yank things down because you played a little bit of a song. On, Sal. Help us out here.
To illustrate this simplified model of evolution, I'll use circles to represent individuals like Mommy and her 3 babies. Further, just for fun, I'll give Mommy the name Eve.
Ah, I see what you're doing there.
I'll also put red numbers in each of the circles to represent the number of harmful mutations in each individual.
And again, we're taking the number of harmful mutations and from just the pedigree mutation rate, where he's assuming that the pedigree mutation rate is the phylogenetic replacement rate, and that 100% of all such mutations are deleterious, and we know that both of those things are wrong.
Yeah.
Okay. And I don't think that can be covered by just saying, well, this is only a pedagogical model. It's not biologically accurate, Sal. I think you need to go a little bit further, because the fundamental basics of your entire idea are verifiably wrong, right? This is just like, if I ask you, hey, how long is my dining room table? And you tell me that it's 42 kilometers long, and I'm like, I feel like it's more like 1.5 meters. That's the kind of wrong we're talking about, right? This is a measured thing. Like, we know there's not a question about this. Now, you might be able to argue with me that it's like 1.52 meters, and I might say it's 1.48 meters. That's a fine discussion to have, but you're in the 42 kilometers kind of range here. You're verifiably very wrong. Based on measurements. That's why I'm using a ruler comparison. I actually don't know how long the dining room table is. I'm just making up numbers, so don't at me about hey, show us the measurements for your dining room table.
With Mommy Eve having 0 harmful mutations to start off with. However, each of Eve's babies has 1 more harmful mutation which Mommy Eve did not have. The reason children were given 1 more mutation than Mommy Eve is that this models Muller's limit for humans. Also,
one.
hypothetically, even though all the babies have the same
Sal, you got to finish your sentences. You forgot to end the
number of harmful mutations, the baby on the lower right corner will arbitrarily be given a harmful mutation that is not as severe as her sister's mutations. To emphasize this, I'll make the font of the numbers different sizes to reflect the relative severity of the birth defects among the siblings. The smaller font will reflect a mutation of lesser severity, and a bigger font a mutation of greater severity.
That is getting needlessly complex in terms of like data presentation, but okay. I also want to say Dr. Tapioca Weasel has gifted one Dapper Dinosaur membership, and it went to let's see, Finn. So welcome aboard, Finn. Thank you very much. And let's see. Oh no, I thought someone else had gifted a membership, but that's just me looking at the wrong chat. I was looking at the stream chat instead of the live chat that I have up. I totally know how to do this, guy. Trust me, I'm like a professional.
Because the baby in the lower right corner has the least severe birth defect, I will call the baby in the lower right corner the best baby.
Okay, that seems fair, actually.
Strictly speaking, the individual with the most genetically fit genome is Mommy Eve.
Again, that's only because we've assumed that all mutations are deleterious, which means that you cannot ever get a more fit genome than your parents. But we know experimentally that is just not true, right? Like, we have multiple evolution experiments in which offspring are measurably more fit than their ancestors.
Yeah, he also can't measure fitness at this stage. I mean, he could say, for example, like if the other two just like died in childbirth, right? Or they died before they reproduced, or like at this present state, like right as these offspring are generated, he can't say which one has the most or less fit. We need to see how many offspring they have or how long they survive to. So like he's not actually being very consistent in the way he's thinking or defining fitness.
Well, I'm going to give him the benefit of the doubt and say that is something we're going to get to, and he's just taking that information that he knows is going to be coming in the future and just bringing it back to this stage. So, which, you know what? Let it never be said that I have not given the benefit of the doubt to Sal.
And since all her kids have one more harmful mutation than she does, genetically speaking, none of her offspring will be as well off in their lifetime as she is in her lifetime. Therefore, I designate Mommy Eve as the true fittest, as she has the most fit genome of the entire population in this hypothetical world. In contrast, Darwin and Spencer mistakenly designate the fittest individual as the fittest of the kids, namely best baby, and not the fittest of the entire population, namely Mommy Eve.
Now that part I'm going to disagree with, right? Because if you do like an actual experiment where you're measuring fitness across generations, which is what we're talking about here, you're measuring previous generations, and if there is in fact a decline in fitness, which you can induce, right? Like you can drive populations to extinction in experiments, we could measure the higher fitness of previous generations, right?
Yeah, that's how you track changes in fitness, right? You would measure the offspring production in the previous generation, and then you measure it in the succeeding generations, and then you can track, and of course, like, you know, collapsing fitness, which would correspond to a collapsing population, and so you would very easily be able to track those things. So Darwin and Spencer wouldn't mistakenly say that that new baby has, you know, the best fitness. They would say Mommy Eve has the best fitness and baby has slightly lower fitness.
Yeah. And for the purposes of this, I'm happy to just pretend that whenever he says Darwin or whatever, that he just means evolutionary biology. Because otherwise, I would have to be constantly going back and saying, well, actually, Darwin's particular writings didn't include this information about this genetic concept, and I don't want to have to do that. So I'm just going to assume that whenever he says Darwin, he is poorly describing or poorly naming modern evolutionary biology as it is studied and done today by evolutionary biologists, like my guest here, whose link is in the description. Go subscribe. We got 99 people viewing here right now. Now, I know a few of you have already done that, so I'm hoping that by the time this is over, that Zach here has at least 80 new subscribers.
Emphasize again, the fittest kid, namely best baby, is mistakenly designated in Darwin and Spencer's convention as the fittest. However,
No.
in reality, best baby is not the fittest in the population. It is Mommy Eve. This happens because
That is not a mistake that is made.
when Muller's limit is reached or violated, Darwin goes bonkers. Thus, instead of designating best baby as the fittest, as Darwin and Spencer mistakenly do, I'll designate best baby as fake fittest. Of course, that's no fault of this poor hypothetical mutant baby that she's called a fake. The blame for that rests on Darwin and Spencer.
Actually, no, the blame on that in your model would be on just genetics, right? If you were right about this, couldn't blame it on any scientists. It would just be how reality works, right? That's like blaming sinking ships on Newton because he discovered and described gravity. It doesn't make sense. Ships sank before Newton. If we never figured out gravity, ships would still sink. It's the weird way to put it, like, oh, this is because of Darwin. Like, no, if this were real, if this actually happened, it would have nothing to do with Darwin.
The important point, however, is that the true fittest is Mommy Eve.
Okay.
In effect, Darwin and Spencer have gotten away by saying one thing, but actually meaning another. Saying one thing, but actually meaning another is a logical fallacy and or rhetorical trick known as equivocation.
I fail to see how there's any equivocation here. I don't even know how to respond to that other than to say, Sal, you have not actually demonstrated any equivocation. You need to do better. I don't even know what the argument is to say that this is equivocation. You could say they're wrong, right? You could say they're mistaken, but I fail to see where there's an equivocation. Now, I think in part that's because he's projecting the equivocation of genetic entropy advocates of terms like fitness onto real scientists. But yeah, your projection is not sufficient for me to say, oh yeah, that's totally them equivocating, or to even have a response other than just say, demonstrate it.
Yeah,
When Muller's limit is...
Sorry.
No, I'm just trying to, I'm waiting for him to make clear what he means by the fittest or the less fit, in a way that's objective, as opposed to, he just said we'll have a worse life. I don't really know how to objectively measure that in an evolutionary or biological context. And so I'm just waiting for him to make clear to us what he means by fitness here.
That actually brings up something interesting, because I had a debate with Sal a while back on Modern Day Debate, back before they became the National Socialist Bar, which is why I'm no longer willing to go on there, is because they've turned themselves into a bar that accepts National Socialists hanging out, and I don't want to be in that bar. So in the debate, one of the things that I drove home to Sal, and he said he was going to go away and think about, but that I currently have not seen any evidence that he has, is that his definition of fitness seemed to be intrinsically linked to the emotional judgments of humans, right? Mm-hmm. So he would say things like, oh, being bigger, smarter, faster, that's what I think fitness is. And I look over here and I'm like, well, what about this mixozoan that is 5 cells big despite being a jellyfish and doesn't even have mitochondria?
Yeah.
I personally find that emotionally less cool than being, a ceratosaurus. But
yeah,
why should nature care what I think is cool? It doesn't.
Yeah. He does this a lot with gene loss, increases in fitness because of things like genomic reduction, and that just seems to just blow his mind that evolutionary biologists are completely on board with that concept. Mm-hmm. Because to him, if the genome is getting smaller, it must be getting worse, right? It has to be the fitness must be going down. So our definition of fitness must just be terrible if we can incorporate gene loss into our models.
What I'm hearing is, let's make amoebas our kings.
It's true. If you think about, if the converse is true, right, that the biggest genome is the best, then the most complicated organism on the planet is Amoeba dubia. Yeah. Right. I think it's 300 gigabases. It's
like 100, it's 100 times bigger than the haploid genome size for humans.
Yeah.
Yeah. Obviously, amoebas are, in fact, the things really created in God's image. They are the ultimate in biological development, and they should be running the world, not humans or dinosaurs for that.
Reached or violated, Darwin and Spencer's idea becomes survival of the fake fittest, not survival of the fittest. In light of this, it's high time to beat the fake out of Darwinism.
Okay. Is this a Trump meme? It is the Trump meme.
Yeah.
Okay, to be fair, this was made a while back.
Let me simplify.
When that meme was a bit more current. But also, oddly enough, Trump features in the thumbnail to this video, and I don't understand why.
Yeah, I don't either.
It's not. This is just a tip to Sal in terms of like just having a channel, right? Your thumbnail should ideally be something that makes people want to click on the thing, which I think Trump actually probably does. Because no matter which side of the Trump fandom, whether pro or anti, you fall on, he is a very clickable person. Because if you want to hear about how badly he's doing, you click. If you want to hear about how great he is, he clicks. However, your thumbnail should also accurately portray at least the basic idea of what's going to be in the video. And that's where it fails. Because other than this meme, I don't know what this has to do with Trump. He probably shouldn't be the face of this video. Now, Heather Kuhn for $5 says, does Sal have any clue that fitness is situational? He sure isn't acting like it. Now, here's what I will say. I know for a fact that it has been explained to him that fitness is situational many, many times. Whether or not he has internalized that information or accepted it, I cannot say. So, my answer that's my answer there. He should know. He has no excuse not to know. I don't know if he does.
The same is true for the context of any like fitness effect up from mutations, right? Like a deleterious mutation or a beneficial mutation is itself context dependent.
Yeah.
Deleterious in one environment is beneficial in the next. There's not some like objective, this is the all-time forever distribution of fitness effects, right? There, as the environment changes, the effects of mutations change as well.
Yeah, that is really important. I mean, you can just see that in humans with the selection for heterozygous sickle cell trait, where in areas that are heavily affected by malaria, sickle cell trait is very widespread because people who are either completely without that allele or who have two copies are less fit in the environment. Now, in any context other than heavy malaria load, that's not a great thing. Sickle cell trait is just overall not a good thing to have, and it's going to be selected out pretty quickly in most circumstances. But in that circumstance, it's great. I would much rather have sickle cell trait in, say, a heavily malaria-infested area than not. Because probably not going to die of malaria, which, in case you guys don't know, you can look it up. Malaria sucks. It's not a fun time. You do not want it. All right, here we go.
And declutter the diagram of some of the details to make the basic point a little simpler to see. Even though there will be a best baby for each generation for the remainder of the presentation, knowing which baby is the best baby will no longer be important.
Real quick, for £2, unverified real B2022, officially not official. AIG is a laboratory of lazy, lumpy, limp love limbs. Thank you very much for all that alliteration.
Therefore, I'll remove that designation. Likewise, there will continue to be relative severity of the mutations between babies for each generation, but that detail, as reflected in the size of the font, is no longer important for the rest of the presentation either, so I'll resize the fonts.
It is easier to look at now.
Warning. I should mention the following scenes contain Darwinian violence.
Look, it's just Eve from the, the Resident Evil movies going and shooting babies.
So now let's watch evolution play out. Here comes beautiful Mother Nature. Isn't she beautiful? But I wonder why.
Oh, maybe that isn't her. Maybe that... I seem to remember this as being like the Mila Jovovich from Resident Evil, but I guess it's not. My bad on that one. That would have been more cool, Sal. You should have used Mila Jovovich.
Mother Nature is carrying a gun.
Oh, no!
Look, Mother Nature just made a little move toward one of the babies with her gun.
Okay. We don't really need Sal's somewhat inane narration here. he's just going to go through and say that natural selection is taking out the least fit individuals in the population, which, okay, yeah, that is sort of what it means to be the least fit, is that you are selected against, right? So, yeah, I mean, it's weirdly long-winded, and it's very curiously animated, and I'm not sure why, but I mean, we get the point, and I'm not going to disagree that the least fit individuals in a population are going to be not necessarily killed, but fail to reproduce at the rate of the most fit individuals in a population. Because that is one thing that I think it gets lost a lot on creationists, is that selection isn't binary, right? It's not like you were entirely selected against, therefore you failed to reproduce and died. Or you did reproduce and good job, now you were selected for.
Baby on the
One of the things that evolution does is like, okay, we're going to use round numbers to make it easy. So like if Zach has 10 kids, but his brother Dan has 100 kids, then relatively speaking, Dan's being selected for versus Zach. And if that's our whole population, we could say that Zach's being selected against because the proportion of his alleles is going down in the subsequent generation. But just binary. Now, granted, those are ridiculous numbers, and that's not how it's actually going to work out in a real population of a reasonable size. But like that idea carries forward. Unverified real B2022 officially not official for £5 says, inspired by your last Jackson with Jackson, AIG is a bag of suggestively shaped non-gummy sweets. Yeah, it was Charles Jackson put his P.O. box up on screen in my last Jackson with Jackson show. So I made sure to tell everyone not to send him bags of human feces, which I'm serious about. Don't do that. It's a really bad idea. You can get in trouble for it. But it was also pointed out that there is a company that will send a bag of penis-shaped gummies to any recipient you want to, so that you could literally send someone a bag of dicks, and they could eat it, hypothetically at least.
They could.
Yeah. I mean, hey, it's free candy. You might as well.
left. Mother Nature may be beautiful, but she's one mean mother.
Okay.
But look, there's one sole survivor here. It's none other than fake fittest baby.
Oh, by the way, Dr. Tetraweasel with dubious officiality for $5 says, Sal make bad arguments like I make alt accounts to play with myself inside chat. Smash that like button. Yes, please do smash the like button. And I'm not going to celebrate your first super chat because you're just Dr. Tapioca Weasel with a different account. Yeah, Tapioca Weasel likes to try to get one over on me by pretending it's his first super chat. I see what you're doing. I notice.
Let us give the surviving fake fittest baby a name. Let us call her Jane.
Okay.
Let's watch Jane grow up. Baby Jane becomes toddler Jane.
Yep.
Then child Jane. Then teen Jane. Then adult Jane.
So you could just skip to Jane becomes a reproductive age. You don't have to go through every single ontological step. You can skip.
Then eventually, Mommy Jane. Nevertheless, throughout the process, she retains the one harmful mutation she was born with.
Actually, I'm going to go ahead and point out that there is a possibility for back mutation. That's a thing that can happen. So if in her germline there is a back mutation, even if we assume that all mutations from the wholly perfect genome of Eve, which again, there's no reason to think such a thing is even possible to exist. Even if it did, if there's a mutation in Jane that leads to some deleterious effect, it is possible to back mutate. So we can't actually say that she's going to carry this through in her germline her whole life to her offspring.
And even if she does, she might not contribute it to that to her offspring. And even if she contributes the haplotype that carries it, it could have back mutated.
Yeah. So I mean, there's a whole lot of possibilities in which this, even if we were to pretend that up till now this has made sense, even at this step, nope, doesn't actually end up following. Now you could say this is the part where we're being somewhat unrealistic, and to be fair, most of the time this type of mutation probably would carry through, but it doesn't have to.
And her offspring will inherit this harmful mutation. Now remember, Jane was the fake fittest in the prior generation, but in this present generation, Now that everyone else from prior generations is gone except for her, she now becomes the true fittest for the present time. However, she is not the true fittest for all time.
Which again is something that maybe not all time, but long-term fitness is tracked through populations in actual studies. It's measured.
Since the true fittest for all time was Mommy Eve. But to reduce visual clutter, I'll refer to Mommy Jane as simply true fittest, bearing in mind the label only refers to the present time, not all time.
Got it.
And so, like the prior generation, the process will repeat, whereby Mommy Jane's children will get one additional harmful mutation.
Strawberry Vein for $10 says, I hate when young Earth creationists try to argue that evolution is bad because it involves death, when according to them, God cursed the entire world with death because a guy ate a piece of fruit once. Yeah, it is it is a little odd that if you're going to say that death is per se bad in some kind of like moral level, that then God being the cause of at least human death under most Christian interpretations, not all, like it does seem like it would then make God bad to some degree. Now, look, I'm sure there's a bunch of various theodicies that get around that, and this is no theology channel. But yeah, it is a little curious that there's so much being upset about death in evolution when death is all over the place in the Bible and places where God's causing it, and that doesn't seem to be all that bad. Maybe it's only when God directly causes the death it's bad. It has to be like a miraculous level of death. I don't know.
Well, and in evolution, it doesn't even have to be like physical death. It's genetic death, right? So like the individual can live a perfectly fine, happy, healthy life and just leave no offspring, right? That's nobody would say that's death. It's just reproductive death. They just didn't leave any offspring. It's a very different thing than like killing them. Right?
Yeah, actually, I made that point in a video that I think is our own early access still, that if you were immortal but sterile, as far as evolution goes, you've been removed from the population. But as far as you as an individual organism goes, you might be having a great time.
Which Mommy Jane did not have, because we are modeling what happens when Muller's limit is reached or violated. Let's fast forward through one more iteration again in detail.
And this is where his definition of fitness just falls apart. Because notice that Mommy Jane had just as many offspring as Mommy Eve.
Which makes her just as fit.
Which makes her just as fit. And so, like, I don't understand if these mutations are deleterious, right? Then the population, then her fitness should have been less than Mommy Eve's fitness, and yet it wasn't. Right. And so, like, and he's going to do this round again. So, like,
we'll just
for his definition of fitness to be useful, we need to actually see the population collapsing. And that's the thing that genetic entropy never actually addresses. They never make a mathematical prediction. So if we know the mutation rate, if we know the deleterious mutation rate, and we know the population size, they need to put together a model that makes a prediction of when the population should go extinct. And like that's all I'm asking for. And I've yet to see them do anything like even remotely close to anything like that.
That was the end of the video, by the way. That's it. That's all there is to that video. I'm very confused as to how this is supposed to be some kind of like big takedown. It's like, hey, you made like innumerable mistakes. The various assumptions that you made to get to your model were all verifiably, laughably wrong. And then he did some cutesy cartoon with a woman shooting babies for some reason. And I'm going to get demonetized for saying that. And, like, that's going to, like, oh, there you go, guys. Take that, evolution. Like, oh, okay. Cool. Pibblepunk for $5 says, apparently Sal thinks, quote, survival of the fittest means if you're buff enough, you become immortal. I laugh, but the thing is that when you look at the debate that I had with him, it does seem like, maybe not literally immortal, but it does seem like that's the case, right? He equates fitness with, like I said earlier, the kind of thing that your personal trainer might say for fitness, which is perfectly fine in the context of going to the gym. But that's a different context than evolutionary biology. Savage Cobra Savage Cobra official per se, spelled wrong. I hate that you guys know which things to say to make me not be able to hold my composure. You guys know me too well. The population collapse will happen soon, just like the second coming. Yeah, it should be happening immediately, right? Like a more effective model would be like, oh, Mommy Eve has like 500 babies. Only one of them makes it, and then that one only has 400, and go like that.
There's two ways you can think about this. If he wants to talk about Muller's limit, then he's talking about the mutation load paradox, and then he's assuming that there is mutation selection balance, in which case a mutation load of 10, we're dead in the water. If we reject mutation selection balance, and we say, well, we haven't actually reached equilibrium yet, and so the mutations can accumulate. We're not actually to that point just yet. In that case, fine, but the population still would be losing fitness and can't be growing in size. And so even if you want to argue that humans have artificially inflated our ability to persist, we've inflated our carrying capacity, I assume other organisms that are growing in size in nature haven't done the same thing. They don't have modern medicine. And so no population, if genetic entropy is true, should be growing in size. It should be incapable of doing so. And yet we see plenty of populations that are either stable or growing in size.
Yeah. And also one thing I want to say is that modification of the environment to increase carrying capacity is actually not a thing unique to humans. Elephants do it. Beavers do it. So if you nothing about humans is all that special except the degree to which they do things. And it doesn't work in humans. It doesn't work in any other organism. In fact, sometimes I've seen creationists mix up extinction vortices, which is not something we're going to get too far into, with genetic entropy. The problem with an extinction vortex... So, okay, back up a little bit. Genetic entropy basically says the more genetically diverse you are, the worse it is for your species, which is the exact opposite of what's actually measured. The more genetically diverse populations are, the less likely they are to be of conservation concern, endangered or whatever. Extinction vortices basically says that when your population is small enough, you cannot really support any meaningful selection. Therefore, actually seriously harmful mutations might end up even fixing in the population, driving it to extinction, because essentially no selection is possible until it's very extreme. And it might be so extreme that the population can't recover. Those are opposites. Pipplepunk for $2 says, ask a young Earth creationist what a perfect genome would look like. Yeah, there's no answer for that. Now, there is one more thing I want to go over, but we'll do this relatively quickly because we've already been going for about 2.5 hours, and I do actually have another thing that I have to get to later. So, in both chat and in some comments, Sal has been saying this thing. He's been saying, genomes decay despite sustained fitness gains, Grauer 2017. Now, he's occasionally said Koos and Grauer, not Grauer, sorry, Lenski. He's attributed this to Lenski. He's also occasionally attributed to Koos and Lenski. I'm assuming I'm saying Koos right. I don't actually know. Sorry about that. Alejandro. I guess it's probably Spanish. Look. Sorry, guys. Now, the first thing I want to point out is it's a little iffy to do things like quoting part of the abstract, which is something we get young Earth creationists doing all the time. They love to quote like the first few lines of an abstract, which, as you guys might know if you're familiar with scientific papers, that's where you set up the problem that the rest of the paper is about solving. So if you don't put that whole solution in there that you have, you're really not representing the paper correctly. This is worse. This is just quoting the title, and not even the title, part of the title. The phrase genomes decay despite sustained fitness gains sounds like a universal statement about genomes for all organisms. When you put the whole thing in, mutator genomes, so not just genomes, a specific group of genomes, mutator genomes, decay despite sustained fitness gains. In what context? In the long-term experiment with bacteria that's the Lenski lab doing the long-term E. coli thing. So we don't have time to go through all of this. But what I want to do is first direct people to this. Now, Zach, you've read this paper, right?
Yeah.
Okay. Now, I have too, but I'm sure that you are much better equipped to understand it. What's being said here? What is the main idea behind this paper?
Let me scroll down to. There's a very good figure.
I'm not seeing. Oh, there it is. Sorry.
Oh, do you see it? Yeah, it was like kind of slowly loading. Do you see this figure now?
I do now. Yes.
Maybe I can make this a little bigger. How's that? Okay, there we go. We can see it good now. Okay, so first thing we need to understand what a mutator genome is. not all of the Linsky long-term evolution experimental lines are mutator genomes. They're just looking at one particular line that is a mutator genome. And what that means is that this specific line has a mutation in its mismatch repair enzyme. And so what that means is that if you can't fix mutations when they occur, because for one, the vast majority of mutations that happen in your genome during replication are actually fixed. So they get fixed by enzymes, so you don't actually pass them on. When we talk about mutations, we're talking about the ones that didn't get fixed, that get passed on. So if you have a mutation in your mismatch repair enzyme, then you're going to have a lot more mutations than what a normal population would have. Okay, so that's what we mean by a mutator genome. And so for one, that's not representative of virtually any natural population. And we can actually see that in this figure. So in each one of these columns, the first column says nature. This is a natural population of E. coli. And then the other two are mutation accumulation lines, and then the long-term evolution mutator line. The nature and what the green and the blue, so the top couple of bars are really what we're interested in. And then the x-axis is the odds ratio. So for nature, basically what the green bar is showing is that you are far more likely in nature to have synonymous mutations than you are to have non-synonymous mutations. And what those means is just that the mutation doesn't change what amino acid is coded for, right? That's a synonymous, so it's the same word, it's a synonym, right? Versus non-synonymous, you're going to be coding for a different amino acid. Generally, that's deleterious. And so when you see the pattern of a strong skew in the mutational spectrum towards synonymous as opposed to non-synonymous, that means selection is purifying deleterious variation. And that's typically what we see in nature. And so that's what you see here. You see that strong bar to the right of the odds ratio, and that's what that indicates. The two blue bars below that, going strongly to the left, are indicating just amino acid changes as a broad category. So you can see you're far less likely to see amino acid changes, far more likely to see synonymous mutations. So that is just purifying selection. That's a pretty typical sign in basically all natural populations. Okay, so that's we need to understand that as sort of the background. Mm-hmm. The two additional columns are showing, as you can see, the absence of that. So the mutation accumulation experiments, the way these work is they take one back to literally a single cell, put it onto an agar plate, and just let it grow. And so all the individuals are descendants from that one individual, that original cell. And so there's very little diversity. And you do this in mutation accumulation experiments because you want to capture the full range of the distribution of fitness effects, i.e. you want to get rid of selection. You don't want selection to be there purging those deleterious variations. So you make the population size basically one. And so when you do that, you can see very little or any at all sign of purifying selection. And that's indicated by having virtually no signal in those first couple of bars, right? Okay. Now, so that's what that's showing. Now, just next to that is the mutator genome. Now, what they did here was something quite a bit different. Instead of starting the cell with just a single line, this is a big population of bacteria. So there's tons of individuals, so you expect selection to be pretty strong. Instead, you can actually see that the signatures of selection look a lot like the mutation accumulation experiments, not like what they do in nature, right? Indicating that selection is inefficient at removing the deleterious variation in this mutator genome. Now, the reason for that, did you have any questions before I say the reason for that first?
No, although we do have to wrap up relatively soon.
Okay, I'll be quick. So the reason for this, the reason why these two things look like this is in very large populations that are haploid, as in the case in bacteria, there's no recombination. So you can't break up. So if you have a deleterious mutation right next to a strongly beneficial one, you can't break them up from each other, right? They're inherited as a unit. So long as the net gain of that beneficial mutation is greater than the loss of the deleterious one, both are going to sweep to fixation. And that's exactly what you expect to see in any haploid organism. So in this case, what they're saying is you get so many mutations, and so many of them are highly beneficial because you're just getting a ton of them, that many are sweeping to fixation, dragging with them all the deleterious mutations. So that's why you don't get to see selection purifying deleterious mutations, but you still see fitness gain because there's so many beneficials that are getting swept to fixation. Now, in sexual organisms, recombination actually breaks up those associations so that you can isolate the beneficial one away from the deleterious ones. And we see this as signatures of reduction in diversity called selective sweeps around regions where beneficial mutations are being selected.
Okay. So I want to see if I can encapsulate the takeaways that maybe we should have for Sal if he were to watch this, which he might. You never know. First, this paper does not apply to organisms broadly, as in all genomes. So acting as if it did either means didn't read the paper and are citing it anyway, which is inappropriate, read the paper but failed to understand it, which means that you are not equipped to have the conversation, or, and I would prefer this not to be the case, Sal, that you have read this paper, understood it, and are misrepresenting it. In my chat, actively lying to my audience. That's the thing I don't want to have to say is happening, and I currently am not there. So, another thing I want to point out is that this especially does not apply to sexually reproducing organisms like humans, which is an area that Sal would really like to apply genetic entropy to. Even if this did support genetic entropy in a particular group of organisms, it wouldn't be humans.
It doesn't even apply in bacteria, to be very clear.
Right, that was
in
where I was going to go next.
There's horizontal gene transfer, right? So like even bacteria have signs of recombination.
Yeah. And my last thing was like, yeah, even in actual real world populations of bacteria that aren't in weird laboratory setups, it doesn't even apply to them.
Yeah.
Right? There is no way in which this paper supports your conclusion of genetic entropy, Sal. And you cutting off the front and back of the title of this paper as if it were just a general statement about genomes is hard for me to take as something that you did in good faith. So, now here's a few, we're going to wrap up with a few more words to Sal, and then we're going to say our goodbyes, and we're going to be out of here, and I'll be over on Chesh's channel, because while I thought that our show was delayed, apparently it's not. So, okay, Sal, you are welcome back in my chat or on the channel after have watched my responses and anything else to which you would like to respond that is mine. If you want to respond to this, you need to be able to tell me honestly that you have watched this video. If you want to do anything else here, you need to at least tell me, truthfully, that you watched the first 3 things. Because here's the thing. You opted to respond to me. In order to respond to someone, it requires that you know what you're talking about and what they said. When you told me that you didn't watch what you were responding to, at that point I have nothing else to say to you directly until after I've watched that. So, I've informed my mods, and this is me stating it publicly. So, you're welcome in the chat. However, from here on out, until you have watched those, the only question put to you will be, have you watched the Sanford series? If you answer anything other than affirmatively, you will be timed out. That rule is in effect until such time that you watch them. That's it. Because quite frankly, it's insulting to me, and to my audience, and to your audience, to say that you're responding to me when you haven't watched the videos. Okay. I am doing this as a courtesy to Sal, because of the 3 responses that I got up to the point of planning this was the only video that had any substantial content in it. I'm not going to sit here and say, oh, DNA folding isn't real. It is. It just doesn't provide any argument against evolution that I can see. I'm also not going to sit here and say John Sanford is an idiot. I don't think he is. In fact, I'm willing to say he's the smartest man who's ever will live. Just doesn't help. Just makes him more of a liar. Anyway, so we're going to take a look at what's coming up on the channel, and then I'm going to throw it over to Zach for any final words he might have. Then we're going to get out of here and go over to Chesh's channel so you can hear me ramble about news items, I guess. So let's see. On the 30th, we're going to be having Kent with Bent. I don't remember what the title is going to be, but it's going to be some pun to do with the Evil Dead. I think at this point I think it's going to end up being Army of Kentness, because I believe the next movie in the sequence is Army of Darkness. Let's see. So that'll be the 30th. Then on the 1st, we're going to have the Sal's Pal Supercut. Now, if you're watching the Supercut counts as having watched the series, so you don't have to click through all 3 of them if you don't want to after this next week. So that'll be on the 1st, and then on the 3rd, we should be having the 10,000 subscribers special, unless I fall below 10,000 in the next week or so, which isn't likely, but it's possible, I guess. If I do fall below 10,000, I will have something else for you. Perhaps some kind of dirge that I play live. Let's see. I think that's about as far ahead as I want to look. I mean, y'all know there's going to be like another Kent with Ben on the 6th. There's going to be a new series starting up on the 8th. But that's that's far enough in the future that I don't want to talk about too much. Zach, do you have anything that you want to tell people about, like what's coming up on your channel or what they can expect from your output or anything like that before we get out of here?
Yeah, just real quickly, thanks everybody for being here. And if you're really interested in any of this sort of thing and you want to like learn more about it or get more into the weeds, you can find my email on my channel. I really like talking about this kind of stuff, and I'm more than happy to like email back and forth with you and explain in greater detail some of these concepts. You can also find a lot of this stuff explained in mathematical detail on my channel. I try to go into all of the pop gen, both on this as well as just in evolution in general.
Okay. Now, I've just been informed from the chat that Sal has already gone live to say that he is destroying everything we have to say in this stream, and that no, he hasn't watched it. So, Sal, you now have to have watched the original Sanford series and this stream in order to be allowed back onto the channel. I've already taken your mod status, and the next conversation I have with you will be after you have watched everything you have purported to respond to but have not watched. That's how that's going to play out. I don't want to hear from you in person or electronically in any kind of live format until you've done those things. But with that, everyone, please go check out Zach's channel. It's got a lot of great stuff. I've watched numerous of his videos more than once because they are worth watching more than once because, quite frankly, they're very dense with information. If you like this kind of stuff, if you like getting deeply nerdy about evolutionary biology, genetics, even in some cases paleoanthropology and paleogenomics, go check out his channel. This man is an actual PhD in the field. Please check him out. He's got great stuff, and I want to see his channel grow and succeed because, quite frankly, we need more like high-level content about genetics and about evolutionary biology and pop gen and all that great stuff. With that, though, we are going to get out of here. Go check out Chessa's channel. If anyone who's a mod could link her channel, that's where I'll be. Or more specifically, if you want to link her actual show that she's doing. And I need to go get over there, like, right now. So, bye, everybody. Thanks for watching, but before you go, I just want to say a special thank you to my channel members and my patrons, especially those pledging $20 or above. Bob Knob, Tapioca Weasel, Klaus Jepsen, Bent Hovend, Kevin Brostek Van Manen, Landon Knoll, Mabbity Babbity, Monkey They Them, San, Denny5252, Eleron Teller, Phil Rivara, Ian Chen, Mrs. Spexinder, Sphincter of Doom, The Venerable Bede, and Scriptic Mystic. As you probably know, YouTube is a very volatile platform, and from month to month my income on the channel can swing wildly. But the people you see on screen are directly supporting so that I don't have to worry too much about that, and the channel can keep going as it is, and perhaps even improving. If you'd like to join this team, there are links below to join the channel or in the description to join the Patreon. The Patreon starts as low as $1 a month, and the channel memberships start as low as $1.99 a month. On Patreon, you can even get a discount for pledging annually. If you do decide to pledge, you'll get access to an exclusive Discord server just for channel supporters, as well as early access to all of my pre-recorded videos, often up to 2 months in advance. Higher tiers will unlock even better perks. Now, if an annual or monthly pledge isn't right for you, but you still want to support the channel, there is a merch store down in the description, as well as an Amazon wishlist, just for me. And if financial support isn't something you can or want to do, then if you still want to help out the channel, please just like and share these videos and make sure you comment on them. It really helps the channel out. Thanks again for watching.